Good morning, ladies and gentlemen. I am Anjana Wala from the Department of Life Sciences, Maharaja Krishnakumarsinhji, Bhavnagar University. Welcome to the webinar on Spectrum of Life Sciences. This webinar has been organized so that all can stay connected to and interact with the scientific community amid the pandemic. The distinguished speakers are going to cover diverse topics that will enable the researchers to find the solutions and new avenues to tackle the emerging problem. The participants are requested not to press unmute and not press present now. This is a request. If you have any queries, please write in the comment. Now I invite Head Department of Life Sciences, Dr. Bharat Singh Gohil, for welcome speech.
Hello. Good morning to all. Namaskar. Warmly welcome to on occasion of today's our webinar, Spectrum of Life Sciences, Department of Life Sciences, Maharaja Krishnakumarsinhji, Bhavnagar, University, Bhavnagar. So we are warmly welcome to all participants, those who are participating today's on webinar, Spectrum of Life Sciences. Honorable Vice Chancellor Dr. Maharaja Chavda, who is a Vice Chancellor of Maharaja Krishnakumarsinhji, Bhavnagar University. Today's expert speakers, Dr. Rakesh Panchal, Dr. Punjani sir, and Dr. Nishitariya. Today we'll deal this lecture, expert lecture, why we taken this today's the title Spectrum of Life Sciences that I want to tell today. Friends, we know that today we are facing a huge problem against the COVID-19. Just one microorganisms fights against the human and all systems. We are forcing to, but it is not a solution today to live on the earth. Each and every systems comes against the COVID-19 today. In this era, we are with physical distance, we are taking this webinar, and it is also immersed the new such directions in digital platform today. Friends, that's why the thoughts comes in our mind. We have to give the one lighten and the corner of the subjects in life sciences like botany, zoology, and microbiology. Friends, I want to tell today, if we are arranging a seminar on physical platform, it becomes very huge practice for this one, and then we can arrange, it becomes very possible to take this one. But today with digital platform, we comes together, and this challenge is given by the COVID-19 today. I'm very happy to tell today, friends, here, 14 states of this country, participants are together here. Assam, Bihar, Tamil Nadu, Andhra Pradesh, Chhattisgarh, Haryana, Himachal Pradesh, Jammu Kashmir, Manipur, Mizoram, Nagaland, Orissa, Kerala, and Madhya Pradesh also. Friends, we are warmly welcoming today's botany, zoology, and microbiology, all three subjects enlightening today. With a recent scenario, COVID-19 has given the challenge to the human beings. We are trying a lot to against the COVID-19, but no known solution today we have. Our social, the pattern actually changed by the just one microorganisms COVID-19. But the shades of botany, shades of the plants, shades of the animals, if now it is a need to see the entire secret, spiritual secrets of the life sciences. Friends, life sciences, it is our umbrella with three all life. It is a science of life. Today, we try to make this spectrum lighten and the entire spiritual reality of the subjects. Together, we want to be give the shades with the experts, speakers today. I hope that. And friends, our India have very nice heritage. Start from Kashmir to Kanyakumari, we have natural heritage, and it is a duty to serve it. Friends, let us see today's our data comes from Gir National Park and lion population, and that is a 7, and it increases as 5 year 29% increases. Friends, we are very happy to say that life sciences has covered all lighten and shades of the subjects botany, zoology, and microbiology today. I'm warmly welcoming to all dignitaries, speakers, and dear participants. And at last, I want to tell my dear all students of Life Sciences Department, thank you.
Our invited speaker, Dr. Rakesh Kumar Panchal, is an associate professor at the Department of Microbiology and Biotechnology, Gujarat University. He is also a coordinator of the PG Diploma in Microbial Biotechnology. Dr. Panchal has over 27 years of professional experience, including teaching, industrial, and administrative experience. A PhD from South Gujarat University. Dr. Panchal has served at Alembic Chemicals and MB Patel Science College before joining the Gujarat University. He has published several papers, more than 27 papers he has published, and he has organized several national and international seminars and workshops. He has participated and he has been resource person to the international conferences as well. Today he is going to deliver his talk on biologics and biosimilars, art of manufacturing affordable drug molecules for our country. May I now invite Dr. Panchal to please deliver his talk. May I now invite Dr. Panchal to please deliver his talk.
Good morning, friends. It is a matter of a great pleasure to me to deliver my talk on biologics and biosimilars, an art of manufacturing affordable drug molecules for our country. At this moment, I congratulate and thank Bhavnagar University and Life Science Department, Bhavnagar University, for giving me this opportunity and to conduct a webinar on spectrum of life sciences, where we spell about life sciences that includes botany, zoology, microbiology, biochemistry, biotechnology. So all the life sciences that clubs together, and it is a consortium which is coming out of all life sciences that is developing certain kind of relief to mankind in the threatening environment of corona, where the world is threatened. You can see each and every person being afraid of a tiny particle called as COVID or coronavirus. We are trying our level best to produce some kind of molecules, maybe a kind of biologics, and try to cope with the damaging effect of this kind of creatures, which are culprit to mankind. In my talk, I'm going to cover the biological molecules, what it is, how to manufacture, what are the problems, and how these biological molecules are different from the chemical drug. And in later part of my speech, I'll cover biosimilars, where a lot of things can be talked about. Every third person is talking about generic medicine, what the generic is, and how it is being manufactured, and what are the advantage of the generic. So we'll discuss certain kind of biosimilars which are exactly synonyms to the generic drug, which is a chemical one. Here we are talking about the biological one. When we talk about biosimilars and biologics, first, let us go through the definition of biotechnology. As such, biotechnology is consortium of life sciences. As Bharat says rightly, that the life science which nurture all the biological sciences. Similarly, the biotechnology, it is the consortium of all biological sciences. Like in 1992 Convention on Biological Diversity of World Health Organization, United Nations basically, and accept the definition of biotechnology way back in 1992, says that technological applications that uses biological system, living organisms, or derivative of these living organisms to make or modify any products and process for a specific use. That is a drug molecule. So that is called biotechnology. We'll switch over to biopharmaceutical. We say that the biotechnology, whether it is involving in pharmaceutical science or not, then we say that yes, the drugs are created by means of biotechnology, where we are applying genetic engineering, where certain kind of derived molecule from living organism, specifically animal cells, bacteria, virus, fungi, yeast, protozoa, where these cells are derived and ultimately we are producing product out of these cells. The product may be in the range of therapeutical proteins like cytokine, hormones, clotting factors, TNF, like insulin molecule. Everybody is acquainted with insulin, like DNA vaccines, and the last molecule where the world is after, and majority of pharmaceutical companies are looking for, that is monoclonal antibodies. Different kind of monoclonal antibodies has been manufactured by numbers of pharmaceutical companies, maybe a kind of global giants, and they are earning like anything. In addition to this, you can have certain experimental modalities like gene therapy, like stem cell therapy, and most recent one, that is the RNA viruses. So you can have an ability to study the structure, to study the sequence, and ultimately you are preparing a molecule which is similar to this kind of molecules in biopharmaceutical. When we talk about biopharmaceuticals, we can say that these are the therapeutic or preventive medicines derived from the cells using our DNA technology. We call it as recombinant DNA technology. The first molecule which has been approved by the authority, that was human insulin. That says that humulin been manufactured from the gene derived from human islets of Langerhans, and it is been transferred to bacterium Escherichia coli, and the bacterium produces protein which is been insulin, just like as human insulin, we call it as humulin. So this was the first molecule which was been approved globally. When we talk about biologic medicines and scenario of this, we find that there are a sizable amount in the range of blood conditioners, that is leuco, neutro, or pancyclopenia, which can be treated using biological molecules. Another one is cancer. You'll find that various kinds of cancers, where you can talk about colon cancer, breast cancer, even blood cancers. You have the treatment for this. Immune system disorders, where rheumatoid arthritis, psoriasis, and Crohn's disease, where you have the solution out of these biological molecules. Neurological disorders like multiple sclerosis, all this kind of problems to human being. These are the problems where the solution is very difficult. Here in biological molecules, we do have the solution for all these kind of problems. You see that more than 400 biologics are in clinical trials. So one or other day or every day, one and other molecules are coming up. They are being approved by authorities, and finally they are being marketed. Ultimately, the physicians and doctors are prescribing these molecules, and the mankind in general that is getting advantage out of this. These molecules include the therapy of cancer, therapy of Alzheimer's disease. This is where you have certain biological molecules. Diabetes, of course, it is the first molecule where the insulin is been came out of the biological system and it is been working like anything. Of course, you have certain molecules which can use in treatment of HIV/AIDS also. And lastly, autoimmune diseases where you have certain biologics which are being useful. When we talk about biologics, you can say that 58% of products are in developmental pipeline. If you see it, then more than 16% of marketed products today, which is available in market, more than 16% marketed products are coming from biologics. And if you see the growth of pharmaceutical industry so far as biologics are concerned, then this is growing at the rate of 20% every year. This is a huge growth. If a company start manufacturing biologics in 5 years, it can reach to the 100% share of the company, where you will say that if it is maybe a 100 billion company, it can goes to 100 to 100 billion within 5 years. 20% growth has been achieved in biological molecules. In 2016, if you talk about the 7 out of 10 top pharmaceutical products, we are talking about top pharmaceutical products, these were biologics. And it is having potential to reach up to 50% share in global pharmaceutical market in next few years. Says that if you go to market and purchase any medicine, you will find that the one medicine out of 2 that comes from biological system. And these are a highly attractive molecule to be manufactured by pharmaceutical company. Of course, manufacturing is a tedious job, but we'll go through it step by step. Where you are going for the manufacturing biopharmaceutical, you'll find that there used to be certain sequence of reactions you are interested in, and you have to do it. First, you are going to identify the human DNA sequence for the desired protein. Any protein in which you are interested, you go and get the DNA and get the gene where it is being located. As you know, that human genome project has been completed. You have all the idea about the gene location on the chromosome. So what you do is you just pick that gene from this chromosome and then after and find out the sequence of DNA of this gene. After that, once that gene is there with you, what you do is you need a vehicle, you need a vector which can carry your gene into some other organism where you expect this gene to be expressed. So various kinds of vectors are available in molecular biology. They are available commercially also. What you do is you have to select an appropriate vector for your gene and then attach your gene to this vector. After that, this attached vector, which is called as recombinant molecule, this is being transferred and you are inserting it into the organism in which you are interested in. So that is to be transferred to bacteria, or maybe transferred to fungi, or maybe transferred to any other microorganism where this organism is producing product for you. And what you are doing is, as a whole, it is called as recombinant DNA technology. Organism which is there with you, transformed one. So what you do is you nurture this organism, you provide nutrient to this organism, and the organism will produce product for you. JDH Helden for the first time told that why trouble to make a molecule when a biological system is making it for you. So he suggested, he directed that when a biological system can produce a complicated molecule, why you trouble to produce this molecule in laboratory? Why you try to synthesize? It is very complicated. It is next to impossible. So what you do is you nurture your organism, where organism will produce product for you. Later on, they have find out some organism to produce some natural products, and then they came to a conclusion that all organisms are not producing all kind of products. There are certain products which organisms do not produce. So what can be done? So after a few years, JDH Helden again told the second statement: if a bug is not producing product of your interest, make the bug to produce it for you. And directed that you can engineer this bug. The bug which is not producing product of your interest, you engineer the bug in a way where these bugs now start producing the product of your interest. This is what the recombinant DNA technology, which is being spelled out of this. The exact DNA sequence is to be known. Type of host cell which is to be used is also to be worked out very well. And then you can come to final stage of product formation. After this, if you go through a sequential steps of this, then the first one is modifying the selected cell. The cell is modified by transferring the gene. After that, one modified cell is there with you, maybe a single cell or maybe few cells. What you do is you are going to grow this cell in a cell line from the original modified cell. So you grow it in mass. After growing it in mass, you go for large number of cell production of this cell line. That says that you can use a kind of vessel, or what is been called as a fermenter, or it is a cell culture vessel, where you are growing, cultivating the organisms, you are nurturing the organism, you are providing all the required facility to the organism, including nutrients, and then these organisms will produce the product for you. So you'll find that the product is produced by the organism in your cell culture vessel. After that, you have to separate this protein from the cells. If it is intracellular, you need to break the cell and get the protein out. If it is extracellular, then the method is comparatively easier. Where it is extracellular protein, you have to separate the cell, and then from the broth, you can separate the protein. After the protein is separated, then the steps follow is to purify this protein in pure form, so as that pure form can be used as drug molecule. When we talk about biological molecules, then one question comes in the mind that how it differs from a chemical molecule. If you talk about small molecule drug like aspirin, it is the smallest one. The chemist can synthesize it very easily. It is low molecular weight molecules. What you need to do is adding and mixing together some known chemicals and reagents in a series of controlled and predictable chemicals. Entire reaction is under your control. What you need is you have to identify A, B, and C reagents. Then after you mix A and B, after it you heat it. Later on the product is formed. To this product you mix the C reagent. After that you can change the temperature, you can change the environment, and then after your D molecule is produced. So here, if you see, you will find that the technique is usually same as for the innovator product. The product which is been innovated by a company, whatever molecule is produced, the same manner you are producing in your laboratory. There's no difference at all. Production process is highly standardized. They are in chemical molecules, and the contaminants in this, the problem creating molecules in this, that they are consistent and they are being quantifiable. Means everything is in your control. So what you do is you have to control all the parameters and finally produce the product. And it is a small molecule, so easily it can be produced. Comparative days, if you go for biological molecule, lot of problem comes. If you find the differences, you will find that so far as molecular size is concerned, your conventional drug is 10 to 1000 Dalton molecular weight, whereas your biologics that starts from 5000 ends to 2 lakh Dalton. It's very large. Since it is very large, the molecular structure is complicated. It exhibits complex spatial structure, and it is difficult to determine the structure also. Whereas for chemical molecule, it is simpler. Simply, the structure can be determined through analytical technique. You have numbers of instruments available, sophisticated. You can put the molecule therein and get the structure of the molecule. Whereas biological molecules is difficult. Complexity, if you find that relatively pure and fine molecules can be produced where it is a chemical reaction. Whereas the complexity of the ingredients is highly complex. The impurities are also difficult to remove. To leach out this impurity is also a problem. And of course, where you produce a biological system, numbers of byproducts are also been producing, and these byproducts are not good for you as a whole. This byproduct is going to disturb at the time of purification. Clinical behavior, if you see that the well understood clinical behavior on mode of action of chemical molecules, whereas the complicated mode of action that is been seen in case of biological molecules. So far as manufacturing is concerned, it is a specific and complex biological process required for manufacturing where all the parameters are to be controlled by you. And if you compare with chemical synthesis, as you know that there is A, B, C reagents, and then you have everything in your hand to be controlled easily. Batch to batch variation, if you find in case of chemical reagents and reactions, you'll find that small to negligible variations are observed. Whereas here in biological system, if you are going to manufacture it, you'll find that no two batches will be entirely identical. So if I'm producing in my laboratory, one batch, the molecule is produced. Okay, after that, when I go for the same batch to be repeated, I'll find that there are numbers of variables and hence everything is not to be mimicked as it is. So it is very difficult. And this is how all the pharmaceutical companies manufacturing the biologics, they are running in a way where every parameters to be controlled is a costly affair, and that is how this company used to market this kind of product at very high rate in market. Manufacturing cost, of course, it is high relatively compared to your chemical molecules. So far as complexity of biological molecules is concerned, I'll give you a very simple example that if you go through aspirin, a chemical molecule, the molecular weight is merely 180 Dalton. So everything is known to you. Every molecule is understood by you properly. But if you compare with biologics, you'll find that insulin, for example, it is 5,800 Dalton molecular weight. So it is quite complicated. If you jump to human growth hormone, of course it is biologic and it has been manufactured by pharmaceutical company, 22,000 molecular weight that is there. And again, the recent molecules which are being marketed by numbers of giant pharmaceutical companies, it is monoclonal antibodies. The molecular weight is 150,000 if you go for IgG kind of antibodies. So if you compare 180 Dalton molecular weight and 150,000 Dalton molecular weight, then you will see that a lot of complexity is there. And due to this complexity, the manufacturing is a problem. Again, to understand this in other way, I can say that aspirin, that is 21 atom molecule, and which is comparable to your bicycle, maybe about 20 pounds of weight, and complexity is less. If you compare this with human growth hormone, that is 3,000 atoms, and it is comparable like a motor car, where you find that 3,000 pound large engine and large kind of complicated machinery. Still, if you go higher, you will find that IgG antibody, what we call about monoclonal antibodies, 25,000 different atoms are forming it, and you have to control over every atom. And it is just like as controlling your private jet, which is 25,000 pounds of weight and highly complicated engine. Complexity is very high, and this is how manufacturing is creating problem. So manufacturing of biologics, that is very difficult to be manufacturing. Stages of product approval and commercialization, if at all you are manufacturing, you need to go through the various stages in pharmaceutical industry. Like product development is done at your laboratory, where you say that you have isolated gene, transfer it to vector, transfer it to host cell. Again, that recipient cell is transformed, and you grow this cell. But once it has been grown in laboratory, then you need to scale up the batches. Says that from maybe 100 ml, you have to scale up to 1 liter, you have to scale up to 10 liter, and then 100 liter and thousands of liters. Again, if you go for all this kind of scale up, you need to have engineering batches. So batch to batch variation used to be there. You need to involve a lot of engineering parameters to be controlled. Then after, once the product is there with you in your hand, you need to go through clinical trial of the batches, where you have phase 1 trial, phase 2 trial, and phase 3 trial. And when your molecule is passing through all the trial, and then you have an ability, or you can get a license to go for validation, and then commercial batch can be manufactured. We are talking about the COVID vaccine. Everybody is looking after the vaccine, and says that numbers of global giant is after this, but still vaccine is not available. And if you find tele news channels, you'll find that every day they are talking about one or other kind of vaccine molecule has been manufactured by some company. But when it has been manufactured by the company and it is very good being shown on TV channels that yes, this is the molecule and this is how it is working, but whether that molecule is there with you in market, the answer is no, it is not in market. What is the reason? The reason is they are going through a clinical trial. So here in India, the coronavirus vaccine has been manufactured by numbers of companies. They have efficacy also, but all these molecules are under clinical trial. And this clinical trial has to be passed. And once the clinical trial is passed, later on, they will go for validation. And finally, after validation, they will ask the government bodies. And then after, once the approval comes, after that, they can market. So everybody is talking about next month or next to next month, or maybe in August, maybe in September, or maybe in October, where the corona vaccine will be available. But still, lot of things are to be worked out. Lot of variables are to be controlled, and then that vaccine can be there to the pharmacist, and ultimately it can come to you where you are being protected against this COVID-19. Biopharmaceutical, when we talk about, you say that strong relationship between manufacturing process of pharma and the characteristic of the final product. If the process is changed a little, you'll find that your product is changed. And when your product is changed, its efficacy may be changed. Not only efficacy is changed, suppose the efficacy is the same, it has not been changed. But if the structure is changed, its side effect may be changed. And that is also not allowed. So manufacturing biologics, that is more complicated. A high level of precision is required to produce the consistent product time after time. You'll find that in pharmaceutical companies, they go for utmost care. Every step of the pharmaceutical process manufacturing, that is being cared by the highly expert people. And this company is giving a very handsome salary to this kind of scientists and workers, and that is how the final cost of the product that comes to be very high. If you find in the pharmaceutical company a small changes in the product is mentioned, you'll find that that product is of no use. Why these changes came? So you say that small changes in the production process, maybe equipment change, maybe environment change, you'll find that the variation is ultimately converted into behavior of the cell, which has been changed. So once the equipment is changed, once you are changing your machine, you are changing some environment where your organism may not be comfortable in another equipment, another machine. So this change in the organism, it changed the protein which is produced by the organism. And when the protein is changed, you will find that three-dimensional structure of the protein is altered. And when the three-dimensional structure of protein is altered, you will find that there used to be poison in the effect. And hence this molecule is of no use. Company has to discard this molecule. Manufacturing of molecule is costly like anything, but still it is to be discarded. You'll find that quantity of acid-base variation, glycosylation, this is a biological process going on in the cell. And you are using the cell to manufacture a product. The cell is manufacturing the product, but at the same time, cell is growing and living. And when it's growing and living, you will find that hundreds of reactions, thousands of reactions are going on. So you are not controlling each and every reaction from your side. And that is how, if your organism is behaving well, you'll find that your product may be all right. But if your organism is, means in second batch, the organism changes. And when it changes, you'll find that all your hard work that is vanished. Again, you say that when the protein is changed by little changing, you'll find that the safety of this molecule, the effectiveness of this molecule, that has been altered. And when it is altered, this molecule is not allowed to market. And hence, the pharmaceutical company has to go through numbers of steps which are to be controlled manufacturing the process. And all these steps are to be corrected in a way. So you find that high quality and consistency of the product, if it is to be manufactured, a biological drug requires around 250 in-process testing. Means during the manufacturing, you have to go through the test, 250 different tests are to be followed. And then your consistency is been maintained. Your quality of the molecule is been maintained. And if you compare this with chemical drug, you'll find that compared to chemical, you'll find around 50 tests for small molecule drug. Maybe if you are manufacturing aspirin, you need to go through 50 different tests during manufacturing process. If you are manufacturing insulin, you need to go 250 different tests. So this amplitude of test, that suggests that this is quite complicated to be manufactured. In the picture, I have shown the fermenters where all the lines are stainless steel lines, and then these lines are carrying one or other kind of molecules to be fed at the time. Where you are fermenting the organism and to produce a particular product, you will find that this organism is to be maintained in a way where you have to care much more than you are going to care your baby. And there, every step are to be precisely controlled. And at what time, what is the concentration, and up to what time it is to be continued, to be added. Then after, what kind of test is followed. Then after, if the little more or less been added, what else can be added, how to control it. So everything, right from first minute to the last minute. In fermentation industry, if you go to produce a simple molecule like penicillin or erythromycin, using the Penicillium chrysogenum, maybe a kind of fungi, or it can be Micromonospora actinomycin, you'll find that these organisms are to be nurtured in fermenter, and they are to be grown carefully. And this growth cycle of manufacturing, that is of as many as 6 to 7 days. So you are going to control the parameter from a minute where your batch is started, 2 minutes of 7 days where the batch is ended. And hence, it is very tedious to manufacture. And where I just memorized where we were producing penicillin and erythromycin in Alembic Pharmaceutical there in Vadodara. There what we were doing is right from first minute to last minute, all the parameters will be controlled. We people used to go on odd hours also to the company. And finally, the product is manufactured. The product manufactured cost, if you go through, the costing can be, say, 1 rupee per tablet, but this 1 rupee per tablet is sold in market at the cost of 35 rupees per tablet. See, the huge profit is there, but why huge profit? Because in 1 rupee per tablet, that is also too costly. If you compare with aspirin, aspirin can be manufacturing cost maybe less than 5% per tablet. So this is the difference where the manufacturing is being done. So if you are going for manufacturing at laboratory level, where you can say here, this one is a control unit where this is going to control your fermenter. And the second one is a complete fermenter where it is a glass window where the fermentation is going on. You do have numbers of probes are connected to this, which control the temperature, the pH, then dissolved oxygen, then anti-foam. Everything is to be controlled automatically through this control unit. Of course, you have a boiler there to generate the steam under pressure, and this steam under pressure is connected through various stainless steel pipes where the steam goes to the fermenter, sterilize the fermenter, and then after keep the fermenter sterile throughout the process. And of course, you need one chiller also to supply the chilled water, which can be supplied in this jacket of the fermenter, whereas the temperature of the fermenting molecule or fermenting organisms, that is to be controlled right from first minute to 5-6 days, whatever your process is. One more time, I would like to tell you that where it is, everything is the stainless steel pipelines and stainless steel valves are there, and batch to batch variation. As I said, that every batch you have to be utmost care, and one first batch is all right. That doesn't mean that my second batch will be identical to that. So every time, every batch, we need to change certain things. We need to control certain things, and that is how the product manufactured is very tedious in case of biological system. This one is a laboratory fermenter, which is of New Brunswick, 5 liter fermenter, where you can charge 4 liter, and then after you start your fermentation, all kind of parameters which I have shown in early case are there being controlled automatically. And this fermenter, where it deals with the product manufacturing for you, where you will find that your organism is nurtured inside and it produces product of your interest therein. One more vessel I would like to show, that is of Sartorius company, USA, and this one is a 2 liter fermenter, and of course all the fittings if you find, that is all kind of the controlling parameters through the probes, and then the 2 liter biological system is being fermented inside. Of course it is a jacketed vessel, and the fermentation is to be done in a way where you control all the parameters. This one is as I say, Sartorius USA made fermenting system. This system do have the control over supply of oxygen inside, where supply of oxygen to us, when we talk about corona infection and we are worried about whether ventilators are there or not. If a person is suffering from corona and if he or she is admitted in hospital, every person is asking whether ventilator facility is there or not, isn't it? So what the ventilator is and why? Say that yes, certain oxygen is to be required to push inside your lungs so as your life can be lengthened. Similarly, here in biological system, we need to supply oxygen inside. And not only we supply oxygen inside, but oxygen tension is also measured through the probe. And then it has been displayed there on this. And it has been controlled automatically in a way where we need to connect the oxygen cylinder along with this. We need to connect the air pipe, where the air compressor is supplying air inside. And at the same time, what we do is we used to connect the oxygen cylinder, and the system itself, what is the amount of oxygen to be added there in the fermenter, and what is the amount of air to be mixed with. So normally where the problem is not there, it takes the direct atmospheric air and supply oxygen there inside. And when the turbidity increases, when inside environment that is not supporting the transfer of oxygen sufficiently, at that time the instrument itself will decide to allow oxygen, pure oxygen from cylinder to be pushed in the vessel. So as my meaning is, your organism is to be nurtured in a way where it is more than the life of a person. Similarly, you have to control all the parameters and grow it in a way where it produces the product of your interest. This is what I have shown, that in our laboratory we used to grow the fungi in form of pellets. This one is 48 hours grown inoculum, which is in form of pellets. It is Aspergillus niger for production of the organic acids. That spherical bodies are to be produced. We used to inoculate the exact number of spores of fungi in the medium. Then we grow on shaker for 2 continuous days. We monitor all the shaking speed there on the shaker. And then after this is been observed, these are the pellets which are small spherical bodies, maybe small bead-like structures, right? So they are the live mycelia. It is the spherical mass of mycelia. Same. Under microscope, if you observe this these pellets under low power objective, you will find that these pellets are the spherical body, where in central it is tightly packed the mycelia, and outside is growing and it is tapering ends which have been shown outside. Again, the same pellets under microscope, but if you change to high power objective, that is 40x objective, 10x eyepiece, this is 400 times. You'll find that in 400 times magnified image, you'll find all the tubular structures, mycelia and conidiophores are being visualized here. All this mycelia in the central portion, which are tightly packed, they are producing product of interest, and the outside flanking mycelia, they are absorbing the nutrients, and it is processed inside and it produces product of your interest, or fermented broth. Now, when we go for manufacturing process, you need to go for cloning and selection, gene of interest, then vector, then host, and all the technology is to be validated. After validating the technology, you need certain cell banking also. There are certain cells which you grow, which has been proved to be best for you. These cells are to be kept in kind of cell bank. and then after, as and when required, you can get the cells in viable form. You need to have warehouse control also in industry, there the input material, output material, that is to be controlled. Manufacturing process you should go for, it is upstream and downstream. Upstream, that is the manufacturing the product of your interest, and downstream is recovering the product of interest from the manufacturer broth. So in case of upstream, you will find that media is to be controlled, inoculum and culturing is to be done, as we have seen in flask where the pellets are formed. Seed generation is done, that is pelleted, the mass is there, this pelleted mass is to be transferred to fermenter. You need a bioreactor or fermenter. That intracellular and extracellular product are being produced in fermenters, and then after once the product formation is there, the product is to be separated by centrifugation. Even if you need to lyse the cells and break the cells, if it is intracellular molecule, that also can be done. So it is in upstream. If you go for downstream, you need to separate the produced product, where chromatography is the best alternative. You can use chromatography, then you need to have a diafiltration, ultrafiltration, and then once the product is separated, then after you need to have formulation. Filtration, of course, 0.22 micron filter are used, so as it is been sterilized and purified, and then finally it is to be stored at low temperature in the company. So all these things are required, and all kind of facility is to be done, and that is how you need that. The molecule is little costly. Manufacturing process, if you go for the filling and packing, then you need to have formulation, then after sterile filtration, then after filling into the container, or you may have vials and different kind of vial sterilization and all this. In this webinar, if the time permits, I may have one or two small video. I'll show you the video where the automatic vial filling machine, where vial has been washed, sterilized, and then after the product has been filled, then after it has been sealed, untouched. No human touch is there. Of course, there used to be various kind of robotic machines are available nowadays, and major pharmaceutical industries are using this kind of machines. Sealing and labeling, and finally packing and dispatch. Right. So once these things are there, your product is produced, it is packed, it is dispatched, and it is going to market. But what about the waste handling? So that is also a big headache for pharmaceutical industry, because when we say that biological product has been produced, a lot of waste material is coming out of this. This waste material are to be treated in a way where it should be safe to dispose outside in environment. So this is also a headache to pharmaceutical industry. Every industry is looking after pleasing the pollution control board people, so as they may not have a closer notice. In case of analytical microbiological testing, there you say that in-process sampling, every time means every 2 hours or 4 hours, you are collecting the sample, analyzing the sample during the process. Then after finished product, then sampling is done. Test method may be a purity, impurities, charge variance, concentration. After that, if it is a kind of antibiotic, you go for bioassay, sterility testing, whether it is sterile or not. And after you need to review the data, release the data. You have to keep the data also, so as it is Food and Drug Administration, which is taking care of all this kind of stability of product in case of real-time stability, and then accelerated time stability is also to be studied. And at last, what you study is self-life of the product, so as the date of expiry has been decided, and you mark this date of expiry to the vials which you manufacture. So far as validation in industry is concerned, you are going to validate everything. I say that right from first molecule which is coming to industry to the final product which is producing and going out of industry, it is to be validated. Process validation, cleaning validation, software validation, equipment, instrument validation, method validation, even your filters are to be validated. Means everything is to be validated, and for this validation, there is a standard protocol which are to be followed. So this is what the manufacturing of the biological molecules, which has been very complicated, and of course it is little costly in market, which has been marketed by the company. If you go through the biotech drug approval by the authority, that is started from 1982 to 2008, you will find that in 1982 only one biological product has been approved by competent authority. And later on, years passed, you will find that 1, 2, 3, 4, 6, like And if you go for 2008, it is 32. And if you talk about 2019, it is more than 150 molecules which have been approved by competent authority allow pharmaceutical company to manufacture. So this is what the biologics are. This is what the biological molecules are, and the molecules are to be manufactured. And right from first day where you think in your mind and the molecule coming to the market, huge amount of expenditure is done by the industry, and that is how industry is getting all this amount back. Not only all this amount back, but making profit out of it. As I said, that 1 rupee manufacturing cost molecule is sold in market at the rate of 30 rupees. And so you will say that maybe 30, 40, 50 time profit. That is the scenario of pharmaceutical industry. As you know that the manufacturing cost, when I talk about in where I say that 1 rupee tablet which has been manufactured, but this tablet has to be marketed. All the marketing force is applying there. You go to physicians, doctors, and all kind of ins and outs of marketing that is been added to this, and it is so that the is in tablet in the market. This is what the journey. And there are numbers of companies who are having ability to mimic this and to manufacture. So what we do is we go through the patenting. Company used to get the patent and patenting. No other company across the globe is allowed to manufacture this molecule. That patent law that is been protection of your right. And that is there for 10 years, maybe 12 years, maybe 15 years. It depends upon the molecule and it depends upon the application which has been done by you. But once the patent is there, then you, till the patent period, you manufacture, you earn the benefit. But after the patent period is over, now what is it? Now this molecule is open for manufacturing. And when it is open for manufacturing by anybody, there you can say that any company can study the molecule,
Sorry for the interruption. There is some technical issue. We'll be again reconnecting short while.
And this is how these molecules are comparatively cheaper to manufacture. So these kind of molecules are called as biosimilars. It is not just like as your generic molecule, because generic molecule, there is a demarcation in between your generic drug and biosimilars, because generic drugs are exactly identical to the original molecule, whereas biosimilars are not exactly identical. So when we talk about biosimilars, you can say that the biological medicinal product
Hello. Yes, again, we will be reconnecting in a short while. We are just resolving the technical issue, so please bear with us. Sorry for the inconvenience. We are just resolving the technical issue and we'll be reconnecting in a very soon. So we are having a very lucid presentation by Dr. Panchal, but just due to the technical issues, we are not able to stream it currently. But just within few minutes, we are going to reconnect, and I'm sure the participants are enjoying the lecture, and where already Dr. Panchal has started right from how the cultures are nurtured in the lab, and he has even led to the fermentation technology and how the product is analyzed, how one can lead to the validation of the product, and the young researchers will definitely be aspiring for their research pursuit in this line. So we are just expecting Dr. Panchal to resume his talk. So I think this technical issue is like coronavirus, very difficult to handle, but we will be overcoming it in a short while. Please bear with us.
So what she has told, friends, Dr. Panchal has actually presented very nicely drug manufacturing. That is, it is our challenges today. Actually, how to we overcome this problem? We are fighting against the COVID-19. Dr. Panchal has explained very nicely that how we, with our energy, we can manufacture this drug. Uh, Microorganisms too. So he has explained the range of description from non-living to living world, how it makes the effect. I think so. Within few minutes, it will solve the problems. If and it is now nearest to time to conclude the first session, and another speakers also will come within few minutes. And I think so many participants has questions and some queries regarding the presentations. But if Dr. Panchal is come online, then we will delete the methods. But before that, I am requesting to Dr. Anjanawala, please to introduce the second expert speakers, Dr. Nishitariya. So Anjanawala, please introduce the second another.
Our next speaker, Dr. Nishitariya, is an associate professor at Department of Life Sciences, Hemchandracharya, North Gujarat University. Dr. Dharaya has 26 years of teaching and research experience. He is a very well-known researcher in the mammalian ecology, and especially he has contributed a lot on sloth bear. Dr. Dharaya has produced over 70 publications. He has completed 14 projects and produced 10 PhDs. He is holding key positions in several professional bodies like State Wildlife Board, Gujarat State, then Wildlife Advisory Committee for Balaram Wildlife Sanctuary. He is also serving as a member in the International Sloth Bear Management and Research Association, and he is he is co-chairing IUC and SSC committee on the sloth bear. Dr. Dharaya has been a recipient of prestigious awards like Booster Research Grant, and he is a very well-known person in the field of, as I said, in the sloth bear research. Recently, he has presented one short documentary on sloth bear as well, and he has delivered several popular lectures and invited talks. So, may I now request Dr. Dharaya to please speak on the challenges and opportunities for research on lesser-known mammals. Dr. Dharaya, please.
Good morning, everyone, and thank you for the introduction. Thank you, first of all, to Bhavnagar University, Department of Life Sciences, especially Dr. Gohil, Bharat Singh Gohil, for organizing such a nice webinar and overwhelming response for this webinar, and it shows the success. So I once again I thank Dr. Bharat Singh Gohil and his team for organizing this seminar, and also inviting me to give my talk on the lesser known fauna of our state. Am I audible? So I can start sharing my screen.
Please. Yes, sir, please. You can.
Yes.
Okay,
Okay.
please continue, sir.
Thank you for your presentation. Thank you.
Continue, sir. You are audible.
Okay. Is my screen is visible?
Sir, your slides are not visible. You may please share your slides as well.
Okay. So let me do it. Can you tell me, is it visible now?
Not yet, sir.
Yes.
Yeah, it's going. Yes, it is coming up. Yeah.
Okay. So is it fine?
Yes,
Because
sir.
now I cannot see anything. I just see my presentation on the screen. So is it fine?
Yeah, it's there. Your presentation is there, sir.
Okay. So
You may please
friends,
continue.
I'm going to talk about the conservation strategy and opportunities of research for the lesser known fauna of Gujarat or lesser known fauna of our country, because many time researchers are interested in wildlife biology. They want to do research in wildlife, and they always go behind the or go after the charismatic species like Asiatic lion, leopard, maybe sloth bear, tiger, or rhino, elephant. But all these species has been researched number of time. Many people have done research, and some remarkable work has been carried out on these species. A lot of literature is available on these charismatic species or large mammals. But the things which are smaller, the mammals which are smaller, or the animals which are small or less appreciated, less studied, are always having a dearth of data. So generally, these animals or these small mammals or small species of animals, they generally are almost understudied, or we can say very less study is available on this. Why it is like that? And it is not for only our state of Gujarat or for India, but it is across the world. If we see across the world, we will find very less amount of research or very less information of and very less amount of data available on these small species. So I just wanted to tell about these species, what is actually missing in about these species, and what are the opportunities for researchers, for young researchers and students to do their PhDs, MPhil, and research on these species, and what are the challenges for their conservation. So this is what my today's talk about, and I would love to have your questions, suggestions for this. And in this entire talk, I just wanted to give you the case study of our study on small mammals or lesser known fauna of our region, Gujarat state. It is very nicely said by Dr. Richard Freeman that if we conserve the big species or large animals, it is not enough to conserve the biodiversity, because entire biodiversity is like a tapestry, and each species or each creature of nature is connected with this biodiversity as a thread. So if one of the thread becomes weakened, or if one of the thread is not addressed, then we may lose the entire biodiversity. And that is why there is a need to conserve, there is a need to research, there is a need to conserve, and there is a need to protect all the creatures of mother nature. Otherwise we may have many issues with our wildlife and forest and our forest. So this is a map of our country showing different ecological regions, ecological regimes. It starts from the Trans Himalaya on the northernmost part of our country, then the Western Himalaya. Then if you go towards the western side, then we'll have the Indian desert, which is the arid and semi-arid region, which includes Rajasthan and Gujarat state. Then we have a very nice central Indian plateau. On the north, we have a northeast region. Then we have Eastern Ghat and the Western Ghat. So we can see here that our country is diverse, has a huge diversity in climate, in geography, topography, ecosystem, as well as in the flora and fauna. So we have studied in a western region, western part of our country, which is Gujarat, and we have studied mostly in the northern part of Gujarat, which is almost arid and semi-arid region. And we wanted to know the status and distribution of small mammals or the lesser known mammals in these arid areas. We also interested to see the evaluation of the threat. What are the important threats to these lesser known fauna or small animals, small mammals? And finally, we wanted to propose a conservation and management strategy, which would be a species-specific conservation and management strategy for these fauna. So this was our goal and objective to study. And you can see here the study area, which is almost the arid region or arid zone of our state. That includes Patan, Banaskantha, Sabarkantha, Mehsana, and the newly formed Aravalli district. So all these among this area, Banaskantha and Patan are mostly termed as an arid area. Mehsana and Sabarkantha is a semi-arid area, and part of the Aravalli district having some very good forest of terminating part of mountain Aravallis. So we have a varied climate, varied diversity in ecosystem, having the desert, sandy desert, saline desert, then dry deciduous forest, and somewhere we have mixed forest and thorn forest in the region. And this indicates that we may have a very nice, very rich diversity of fauna and flora in our region. So what we have done, we have divided our study area into different ecozones like dry and deciduous or mixed scrub forest, arid saline desert, dry subtropical forest, and mixed forest or semi-arid land. So what we have done, we have divided our entire study area of these 5 districts into different ecozones, and we have studied all the ecozones separately by the transect method, by the sign survey, and even by the camera trap survey to see the status and distribution of the small and lesser known fauna. Looking to the study area, most of our study area is the saline desert, where you can see nothing, miles and miles you can see nothing, only the sand dunes or the saline land or saline landscape. But along with this saline landscape, we have a beautiful forest with good river like Banas and Sabarmati. So we have a good forest around that area. We do have some good wetlands in our region, and we also have the dry and deciduous forest of terminating Aravalli mountains in our area. So we studied all these area according to our stratification of the area. And our main methodology, main approach of our study was the direct count, sign survey, and collecting the secondary information from the local people, forest department, frontline forest staff, and even from the registers of the forest department. So we have stratified the study area, then we have done the sampling by our field survey, and then finally we done consultative meetings with the forest department and the local villagers to analyze the threats on the distribution of mammals in this area. So these are our ecozones and their political boundary like districts. We have dry forest in the Banaskantha and Sabarkantha. We have dry tropical forest according to Champion and Set. We have dry tropical forest in Banaskantha, Sabarkantha, and Mehsana. We do have urban lands and mixed forest in Mehsana and Patan, and we have a lot more land as an arid and saline desert in Banaskantha, Patan, and Mehsana region. So this is what our study area and the habitat type. And if we look at the occurrence of mammals according to their feeding habit, this pie chart shows that most of the mammals that we have, small mammals we have, are the carnivore, then followed by the granivores and the others. So this is the distribution of mammal or classification of mammalian species, particularly lesser known and small mammalian species in our area in North Gujarat region. We have further divided them into the family, genus, and species to know their taxonomic status. And if I look at the arid region, we can say we have 24 species of such small and lesser known mammals in our arid region of our state. These are the list of the small and lesser known mammals. We have not studied sloth bear here. We have not studied the large mammals like sloth bear or wild ass, wild boar, blue bull or leopard. But our focus was on the medium sized to small mammals, and particularly for the mammals like small Indian civet, common palm civet, grey mongoose, ruddy mongoose, but we do have data on the Indian fox, desert fox, grey wolf, hyena, different type of cats like jungle cat, desert cat, rusty spotted cat, and even pangolin and porcupine. So we have collected the information, secondary data, as well as we have collected the field data like sign and sketch of all these animals, and we have focused on these three animals. So this chart shows the abundance of these species according to their occurrence, based on their occurrence, and we have divided into the abundant, common, rare, and very rare. So you can see here the most of the mammals are considered as a very rare species, like 48%. So almost half of these species are very rare in the area, and then it follows by the common species, and then rare and the abundant species. So this is based on the occurrence or based on the occurrence of their data in our science survey or in our transect survey. So this is the sign index or sign encounter rate of different animals or different small mammals that we have studied. And based on this, we have found the rare, common or uncommon animals, uncommon mammals in our area. So I'm just going to focus in further slides, in next slides, I'm going to focus on the small Indian civet, common palm civet, grey mongoose and ruddy mongoose, because these are the animal species or mammalian species which is least studied or almost understudied in our area. This is the result of our threat analysis, and as everyone knows, the major threat to the habitat in North Gujarat is mining and industrial development, which is followed by tourism and pilgrimage, and then it is followed by the livestock grazing and the other threats. So this is the threat table, and based on the threat table, we have done our further analysis. So these are some pictures of the threats. You can see here the grazing in the forest landscape, the degradation of the forest habitat, tourism, pilgrimage, because our region pilgrimage, Ambaji temple, like Gabbar Hill and Ambaji temple is the biggest tourist and pilgrim place. Similarly, the temple of Kedarnath in Jessore, it is also very well known for the pilgrimage, and lot of religious people from across the country, they are visiting this area. You can see here the road kills of two civet. This is the Indian civet, small Indian civet and common palm civet. This is we found during our survey as a roadkill. So these are all the threats on the habitat. And based on the threat, you can see here we have classified the small mammals based on the threat. Like the mammals which are living at the minimum threat, they are half of the mammals, but almost 30% of the mammals, they are living on a high threat region, or they are living with the high threat, and 14% of them are living with the low amount of threats. So what I just wanted to tell you from here, so this is what the basics of our findings, of our results, what we have done, the study in 2008-2010 on the small mammals or the lesser known fauna of the North Gujarat. And then I just wanted to shift on my talk uh, for the current topic on the opportunities and conservation challenges for small and lesser known fauna, and what are the different type of research that students can start or we can do to conserve or to highlight our small mammalian species. So let us start with the first such species, that is Indian civet, small Indian civet. And you can see here, small Indian civet, the global population, according to the IUCN, the population is stable. And this is the list of the additional data that we have. And if you see additional data like decline in number or the mature individuals, we don't have any data. Extreme, the fluctuations, again, we don't have any data about that. Extreme fluctuation in the subpopulation, again, there is no data about small Indian civet. Population is severely fragmented, yes or no? So again, there is no data. So you can see here that almost we are at the dearth of data in case of small Indian civet. We go many times in the forest to see leopard, to see sloth bear, and we also come across this animal, like small Indian civet. We take pictures, but we do nothing. We have never seen this animal or seen this species in as a researcher point of view or as a conservation point of view. And if we ignore such animal species, then the time will come that we are again having loss of this species. Like हमको पता भी नहीं चलेगा और ये this species may disappear from our region or our ecosystems. So what are the different threats? This is the distribution of small Indian civet. Why it is understudied or why nobody wants to study this animal? This may be the reason, because it is omnivore. It is found everywhere. You can see the entire country, or in the Asia, Eastern Asia, you can see the small Indian civet is almost evenly distributed in the area. But the current population status is stable. There is no change, no decline, no increase in the population. And maybe the reason is people may not have the enough amount of data. What are the threats that we have studied during our studies? The harvest of skin in China and other countries. So people are not from Gujarat, but from other areas of India or from other countries. They are poached and they harvest for their skin. Civet musk is another thing which is extracted from the perennial gland of civet, and they are they are sold in the countries like India, Thailand, China, and other countries. And for this particular region, this animal is killed or poached. This is also second most commonly traded civet in the countries like Java, Indonesia, where there is a craze of having civet as a pet in their houses. So for that reason also, there is a known threat on these, and because of all these reasons, the hunting of civet is now increasing in the areas. Even retaliatory killing, there are many misbelief or the myths behind the civet. Many people, even in our region, in our Gujarat state, many people are having the myth about the civet. Many people knows that they are cats or they are carnivore. They also harm their cattle, and because of that reason, whenever they see the civet in their farm or in their around their houses or in their backyard, they generally kill those civet. And almost loss of habitat and change in the land use pattern, it also a threat to this species of mammal. So again, there is a need to study all these things. This is the listed threat, maybe during 2005 2008 time, but there is no detailed study has been carried out on this species, even on the threat to this species in our country, in our state, and in across the world. So there is a need to study the distribution, the habitat requirement for for this species, as well as the current threat and reducing the threat on this species. So this is what the current need of the time, of the hour. Second such species is a common palm civet. Uh, Common palm civet is also equally found in our areas, maybe in Gujarat, Rajasthan, and other areas of our country. According to the IUCN, the population trend is decreasing, and most of the additional data is not available with the IUCN, and that is why there is no red list assessment can be carried out by the IUCN on this species also. You can see here the distribution of palm civet in the world, and again it is found in almost all the states of our country, India, Sri Lanka, Bangladesh, and the Southeast Asia. So again, the species is widely distributed, but the population status is decreasing according to the IUCN. What are the threats? Again, the important or the main threat on this species is hunting, because for the meat, wild meat or the bush meat, people in Northeast India, China, Laos, Vietnam, Thailand, they generally take this species as a feast. They eat this species, and for that, hunting and poaching is very common in this area. There has been a recent great increase in the number of captive civet. So people also want to have this civet also as a pet in their houses. There is a large international trade of both common civet, Indian civet, as well as the palm civet, and they are sold in other part of Southeast Asia, mainly in China, for different regions. And the another important threat that we have found is hybridization. Like there are two type of civets, they coexist in the same ecosystem, and there is a threat of hybridization on this animal. This is a very beautiful animal, but nobody is looking at this animal. Nobody is doing research on this animal. Nobody is doing any scientific activities on this animal. We have a lot of picture of this animal. This is Indian grey mongoose, widely distributed, and IUCN has said that the population of Indian grey mongoose is almost stable in all the countries or in the world. So this is the distribution of mongoose. It is mostly found in our country, then Pakistan, Afghanistan, Iran, and other part of the Persia. Population status is almost not known or stable according to the IUCN Red List assessment. And the threats that we have assessed during our assessment is the roadkills, negligence, myth, and poaching. These are the important threats on the grey mongoose. We have different species of mongoose, and many of you have come across when you are in the field. You have seen different species of mongoose. We have grey mongoose and we have ruddy mongoose. So both the species are there, but we have never tried to understand what are the niche separation, or what are the common niche, or what are the sharing of resources, and how they are coexist in the same ecosystem. So we have never tried to study these things, and the species are getting disappeared from many of our villages, rural roads, and forest areas. Mongoose are also captured and sold as a pet. They are also captured and killed for their skin, and even local market of Nepal, Tibet, China, even in the Northeast India, I have seen people are killing mongoose and selling mongoose for different reasons. Some peoples or some tribes in northeastern India and in Myanmar, Bangladesh, they also eat the meat of the mongoose, and the skin or the fur of mongoose is used for the paintbrush, and for that reason they are killed or they are poached from the forest areas. This is the another species of mongoose, ruddy mongoose. It is native to India, Nepal, and Sri Lanka, and that is why it is very important for us to conserve, to protect, and to study this fascinating species of mongoose, because they are native to us. And again, this is not found in other countries of Southeast Asia, so no research has been carried out in India, Nepal, Sri Lanka, or Pakistan, and this is the reason why IUCN doesn't have the population status. You can see a very restricted distribution of this animal. Ruddy mongoose is found in our part, northern Gujarat, then central Gujarat and south Gujarat. It is also found somewhere in Saurashtra region also, but no data has been available. So if IUCN doesn't have the scientific data or scientific information, they will never show on their distribution map. And that is why you cannot see its distribution map. And this is the reason that IUCN doesn't have the current population status. So nobody knows what is happening with reedy mongoose. Nobody knows whether their population is declining, whether people are killing them, whether they are lacking their habitat or they are losing their habitat, or what is happening with the population. So there is an emergency or alarming need to do some distribution kind of research or threat analysis for this particular species. So again, the threats are almost same with the other species of mongoose. There are local threats, and again, threat analysis not been done by many scientists because it is found in very restricted region. But they are harvested to some extent in much of the Indian region for the fur medicine, so-called medicine. But there is no strong suggestion that this occurs at level of sufficient reduction or population reduction is there or not. So we really have very least amount of information about this species. This is again native species of our country and Pakistan. This is a species or kind of the hedgehog, but this hedgehog, if you see, the ears of this hedgehog is different than the hedgehog that we found in our backyards. This is long-eared hedgehog, very small population or restricted population is there again. This hedgehog is again native to India and Pakistan, and very least amount of study has been carried out on this fascinating species. Here you can see the distribution of the species. It is found in almost in our state, Gujarat. If you go up northward, it is found in Rajasthan. Then it is distributed in Pakistan and some areas of Pakistan and Afghanistan. So again, population status is not known. Nobody knows, right? Because there is no study, there is no scientific information, there is no authentic data about this species, and this is the reason why IUCN doesn't have any population status, right? So when we study the animal in detail, when we have the detailed study on this animal, at that time we may have the idea about this species. Otherwise, the species will disappear. It will get disappear from our country, from its range, and nobody will knows. पता भी नहीं चलेगा और ये species हमारे हाथ से निकल जाएगी. So there is an again urgent need to study these kind of animals. So from Narayan Sarovar Wildlife Sanctuary, Desert National Park, and other areas of India and Pakistan, it is recorded, but there is no detailed survey has been done, and there is no detailed information about the ecological or the anthropological threats on this species. So there is no major threat on this species, and it shows that there is a need to study the species. So friends, what is the biggest challenge? Why we have seen no everywhere when we are talking about these lesser known fauna or the lesser studied or understudied mammals, small mammals? The biggest challenge is no. We don't know information, we don't have information, we don't know many things about them, we don't know their ecology, we don't know their feeding ecology, we know nothing. So what we are lacking the information about these species. We don't have the baseline data to show the distribution map. We are almost lacking the scientific research on these animals, these species of mammals. There is no attention by the researcher, no attention by the policymakers, and no attention by the local community. So these are the three important stakeholders for the wildlife. If we want to conserve the wildlife in any ecosystem, either it is an urban ecosystem or forest ecosystem, or it is a big landscape like a country or state, if we want to conserve our wildlife, we have to involve three important stakeholders. The first important stakeholders is the local community. Second important stakeholder is the researchers, scientific researcher, and the third stakeholders is the policymakers. So when there is no research from the researcher, there is no baseline information from the local community, and there is no attention by the policymakers, we are always lacking the data. We are always lacking the information about this. And that's why there is no important or scientific authentic documentation on these kind of species. Why there is no research? Why there is no attention? Why there is no documentation? Because there is no fund available for this species. A lot of fund is available for tigers, lions, rhino, elephant, because they are bigger species, they are umbrella species, they are charismatic species. Many people wanted to work on it, and that is why most of the funds are flooded towards those species. And that is why very less amount of fund available for research, for attention, for documentation, and to generate the baseline data. And that is why generally researchers, students, or young researchers, they don't want to study on this species because they will never get the fund, the projects on this species. This is about the few of the species that I know about, and I wanted to share my knowledge with you. But there are many other species that needs the attention by the researcher, by the scientists, and by the policymakers. These species like pangolin. Again, we don't have scientific information. We have many pictures, we have many photographs, because people are going in the forest to see tigers or lion, and these all species are like a byproduct. मिल गया तो फोटो खींच लिया, मिल गया तो एक छोटी सी नोट बना के पब्लिश करने भेज दी. But there is no specific, systematic and exclusive research study is going on in such species. So in list of these species, we can add pangolin, we can also add Indian rattle, honey badger, we can also add desert fox, desert cat, we can also add rusty spotted cat, we can also add porcupine, and many rodents like squirrel, palm squirrel, flying squirrel, giant squirrel. So all these rodents are also least studied or understudied in our country or in our state. So let us think to develop some study. Let us think to develop some joint projects so we can have a common platform. We can have a common platform where we can do the where we can give our data, where we can give the scientific information with their occurrence, with their status, and finally we can come with a project. We can come with a document, a collaborative document among all of us, all the university scientists, all the scientific institutions, all the researcher. जब भी हम फील्ड में जाते हैं, इसको देखते हैं, इस एनिमल की साइन देखते हैं, just record the sign and we can assemble all the signs and all the data on a common platform. So we can come up by the 1 or 2 year, we can come up with a lot of amount of data by which we can show the status distribution and we can study the current threats to these species, and we can give our share from the science fraternity. We can give our share for the conservation of such wildlife. So these are the conservation opportunities according to me. Like there is a need to develop the monitoring methodology for small mammals. There is no such monitoring methods available for the small mammals. So the forest department or the forest field staff, when they are going in the field, they don't have the methodology to monitor such animals, and that is why these animals are missed out from our vision. We have to organize specialized training for the forest staff, specialized training for the research students or the new students who are coming up, master students and PhD students who are coming up. We should encourage them to start study on these animals, study on these species. We should also train them for studying these species. There is a need to protect the habitat integrity for these animals, because we never know what kind of animal, what kind of habitat is required by this species. So there is a need to study this. There is a need to restrict the transportation and transportation around the sensitive area, because these animals, they are smaller, and I have seen, or many of us, we have seen the road kills of these animals. So there is a need to do this campaign also to save this animal from the road kills. Being a researchers, we can do the ecological studies and monitoring. We can also study the resource utilization pattern between the sympatric species. We can also study the hybridization, like how jackal and foxes are getting hybridized with the stray dogs, or how the stray dogs are infiltrate into this population and there is a chance of hybridization. how hybridization taking place in the sympatric species, which is found in a single ecosystem, how they are sharing the resources, how the niche is separated of these species. So there is a need to do the research on all such things. We definitely need to have a baseline data, baseline survey on these lesser known fauna. There is a need for identification of critical areas. What are the areas where these species are still flourishing, and what are the areas from our state or from our country from which the species are declining or started declining. So we can identify this critical area, and then we can suggest to the government and policymaker that these areas to be conserved or these habitat to be conserved for such a small mammals or small species. As I told you earlier, we can prepare the database using the eco-informatics. Now the GIS and eco-informatics is flourishing. Everyone knows GIS. Everyone wants to study GIS and ecosystem or eco-informatics. So if we have the data with the temporal and spatial information, we can put it on a common platform. Like we can have a Gujarat small mammal study center, maybe at Bhavnagar University, like Dr. Bharat Singh Gohil can take this initiative as a fruit or as a result of this webinar. We can start the common platform to collect the data about small mammal species, and by the end of the year or after 1 or 2 years, we can come up with the status and distribution map without spending any fund, without getting any fund from anywhere. We just go in the field. Whenever we go in the field, we can collect the information and we put the information on a common platform. And then we can use the GIS and eco-informatics to prepare or generate the maps. So this we can do, and I don't think we need to give more attention or more fund for this project. This project can be a self-sustaining project, but the results of this project would be awesome. That would be great for the conservation of these small and rare mammals of our state. So I have these things from my side. I wanted to share these things, and I would encourage these new researchers or the young researchers to do their studies. Don't go behind the large mammals or charismatic mammals or fascinating mammals, but also study these mammals which are small, lesser known, and almost unappreciated by the researchers, by the policymakers. I would love to have the questions and suggestions from you, and if you want to write me, you can write it on my email. The email is already displayed on this slide. And once again, I thank you all. Thanks to my seniors, my colleagues. Thanks to all the students for your listening. And once again, I thank the Bhavnagar University Life Science Department and the team of Dr. Bharat Singh Gohil for inviting me and giving me opportunity to share my knowledge with you. Thank you.
Thank you very much, Dr. Dariyaji. Really, the session was very informative regarding wildlife conservation as well. So many research scholars and youngers are very eager to make the research insights, but they have no very basics, the guideline for and some. But you indicating very well, sir, Dr. Dariyaji, that you sent the message to all researchers that lesser known mammals are very, very important because of the ecological cycle. If you want to be maintain sustainable development by the force of government policy, and then it becomes very important to take the very baseline database of our country, start from all geographical habitat. So nicely, really you presented, you ignite all participants and researchers. So thank you, Dr. Dariyaji, and we will think in that direction. We can put the very baseline database from all regions, all university, because we have youngers and young research scholars, and we can do very easily that, and we will contribute this station. Thank you, Dariyaji. So I am requesting to Dr. Anjana Wala, please introduce the third person who is a botanist. So I want to say, tell about this botany. Without plant kingdom, we can not grow away, because all animalia are actually very dependent. When the first evolutionary aspect start from that, the plant kingdom, the flora, and that's why I am requesting to Dr. Anjana Wala, third speaker, Dr. Punjani ji, is here. And please introduce Dr. Punjani Anjanawala, please. Thank you.
Thank you, sir. So, our next speaker, Dr. Bhaskar Punjani, is an associate professor and in charge principal of Srimati Panchal College, Kalur. Dr. Punjani has nearly 30 years experience of teaching and research, and he is a PhD in botany, as already said. He has made a significant contribution in the field of ethnobotany, and for that he has been awarded special award as well. Dr. Punjani has produced over 40 research papers and 2 books, and he has produced nearly 8 PhD students. I request Dr. Punjani to please deliver this talk. Dr. Punjani is going to speak on indigenous knowledge on plants. Please, sir.
Thank you, madam. My dear friend, Professor Bharat Gohil, two speaker, Dr. Rakesh Panchal and Dr. Daraya. And just I have seen that Dr. Bhupendra Singh Jadeja is also with us. I must remember Dr. Jadeja and all other senior any colleagues which are enjoying this webinar, all these participants. First of all, I must thank Dr. Bharat Gohil that he has provided this opportunity to share my view with this audience as a botanist. So thank you, sir, Gohil sir, and your team also, organizers. Now let us move to our topic, and I'm presenting now. I'm sharing my this screen with you. Can you see this screen? Hello. Hello.
Yes, sir. Yes, sir. Yes, sir.
This screen having this presentation. Can you see properly?
Yes, yes. Yes, yes. Fine, fine, Punjani.
Okay. Thank you. So may I start now?
Yes, sir. Yes, sir. Can do, sir.
Okay. See, this I will talk about the indigenous knowledge about the plants. Now, when we are thinking about the indigenous knowledge, then we must have indigenous people or the community or the indigenous society, and they are using plants. And once they are using plants, means in the nearby area or their surrounding, there should be forest. So forest is there, means there should be a plant diversity. So I will discuss on the basis of what is there in the world, what is scenario in the India, and finally discuss about what is indigenous this knowledge. And you will be surprised to know that nowadays all this Western world which is behind this indigenous knowledge. So now it is recognized by the world.
Sorry,
And that,
Punjani sir, for interrupting you, but can you please make your presentation as a slideshow?
yes, just now I'm putting. No.
Sorry, sir, for interrupting you. Please continue.
Is it okay now?
Yes, sir. Yeah.
Okay. So under this situation, this indigenous knowledge system, which is very important nowadays, and now if you just look during this COVID-19, that all are behind Ayush, means Ayurveda, that how to gain the immunity and how to come over from the COVID-19. So our indigenous people, they are always getting this. Okay. So these people, they get their immunity, they fulfill the requirement in living in the forest and living in the remote area. So they always play with the or live with the nature and natural products. So before I start, I must uh, dedicate my work to the real ethnobotanists, that is all tribal informants, those who have given information to me on which basis I have prepared. Today's our topic is indigenous knowledge about the plant or on the plant. You know myself, Dr. Bhaskar Punjani. Now see that the world's top biodiversity rich continent is America. And this America continent having 7 mega diverse countries. And our Asia, which ranks the second with 5 mega diverse countries. So India is one of the mega diverse countries. Africa is next, that is third number with 3 mega diverse country, and Oceanic rank number 4 with 2 mega diverse. See, I want to show you that there is a rich biodiversity in the world, and our Asia and especially India is having also mega diverse country. So we must proud for that we are living in India. These mega diverse countries are nothing. These are the group of nations that contains more than 70% of the Earth's biodiversity. And as per the CI, that is Conservation International, to promote the awareness for biodiversity conservation among the world nations. Generally, they have identified this kind of Earth biodiversity in the form of mega diverse countries. So there are total 17 those nations which are mostly located in the tropics and subtropical areas or the region. And this group countries have less than 10% of the global surface, but support more than 70% of biological diversity on the earth. So these 17 countries being identified, representing more than 2/3 of all diversity in the tropical and subtropical area, which includes not only plants but the animals and other systems also. And from this, we have in India biodiversity hotspot, and most of the biodiversity hotspots are located in the tropics. These are the biogeographical region with a significant reservoir of biodiversity that is under threat by the humans. And these countries have less than 10% of the, as you know, global surface, but even though they support 70% of the total biodiversity of the earth. Now, when we say hotspot, means it must contain at least 1,500, means 1,500 species of vascular plants as endemic, and they have high number of endemic species. And each hotspot faces extreme threats by the human, that is anthropological pressure is day by day increasing. This biodiversity hotspot we have in India, there are two region, one is Himalaya and other is Western Ghats. So these are two biodiversity hotspot in the India. Himalaya, you know, on the northern fringe of our country, we have this Himalayan region, that is hotspot, which is world's highest mountain, which includes Mount Everest also. And this mountain rise abruptly, resulting in a diversity of ecosystem that range from alluvial grassland and subtropical broadleaf forest to alpine meadow above the tree line. So we have variety of ecosystem in the Himalaya, and we have another kind of hotspot in the Western Ghat. And this Western Ghat is also known as Sahyadri Hills, which are formed by the Malabar Plains as well as a chain of mountains running parallel India's western coast. And it stretches for 1,600 kilometers from the country's southern tip to Gujarat in the north. So that's why we are also interested to discuss this Western Ghat, because on the north, we have some part of the Western Ghats. Now, before I discuss about the indigenous knowledge, I'm very much interested to show you the different forest types of the Gujarat. Just I will discuss with the help of Gujarat that we have total 4 kinds of this moist tropical forest and dry tropical forest also. Tropical moist deciduous forest is generally located in the southern part of the Gujarat state. These are the different subtypes also that I don't want to discuss in detail. We have moist thick forest, slightly moist thick forest also, southern mixed deciduous forest, southern secondary moist deciduous forest also. Then we have another littoral and swamp forest. As you know, that this Gujarat is very lucky that it's having this 1,600 kilometer seashore. And due to this, we have a different kind of ecosystem, marine ecosystem we can observe. So we are lucky, especially being a Gujarati. So we have littoral and swamp forest on the coast of the 1,600 kilometer this length. We have very good mangroves in Kutch and Jamnagar. Not only Kutch, Jamnagar, but other area also. But in Jamnagar, we have National Marine Park also and sanctuary area. Then we have tropical deciduous forest in most of the area of the Gujarat, which includes Saurashtra, then some part of the majority part of the North Gujarat and some part of the central Gujarat also. Then we have tropical thorn forest, which is mostly located in the Saurashtra region and Kutch, and some part of the North Gujarat, Sabarkantha and Banaskantha. We have tropical thorn forest. These are the different subtypes. See the euphorbia, pure euphorbia scrub forest in Gir and Saurashtra. We have dry grassland. We have weedies in the Saurashtra also. Then we have very pure patch of the Enogenesis pendula forest in the Banaskantha. Boswellia forest and dry deciduous forest. Then saline, alkaline scrub, savanna, saline grassland. Then dry bamboo breaks at some places in the South Gujarat as well as in North Gujarat also. Then southern dry tropical riverine forest, desert thorn forest, Ziziphus scrub forest is also seen along with the euphorbia. Then pure acacia Senegal species are also found. Now floristic diversity. You understand the diversity means different kinds, no? Different species, different subspecies, different varieties. So under this situation, hello. ના હું છે ને એક લેક્ચર આપી રહ્યો છું અત્યારે ઓનલાઈન. ના 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 હું છે ને ત્યાં બધા બેઠા હતા ને એટલે બારોટ સાહેબ ને હું એક લેક્ચર સિરીઝમાં હું અત્યારે વાત કરી રહ્યો છું એટલા માટે. ઓકે ઓકે હાજી હાજી ચાલુ જ છે. હાજી 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 ચોક્કસ ચોક્કસ. ઓકે 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 થેન્ક યુ. आज 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 ओके ओके हाँ सारू 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 ओके 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 चलो ओके आओ. Sorry for interruption, dear friends. Now see Acacia nilotica we know subspecies indica જેને આપણે બાવળ કહીએ છીએ in Gujarati દેશી બાવળ. But we have again Acacia nilotica subspecies estrogens. See the pod, the different pod is there. So this kind of diversity as a being a botanist we must observe, and observation must be very clear, so one can easily distinguish the different species, different genera also. Then we have nilotica, again nilotica, but subspecies Cupressiformis is also there. If you just look, it's just like Cupressus of like gymnosperm, it's growing straight. Then see the Abutilon indicum, જેને આપણે કાંસકી કહીએ છીએ, we call કાંસકી generally, Abutilon indicum, Malvaceae. But again different kinds of Abutilon is also there. Abutilon glaucum, then Abutilon ramosus. So this kind of diversity, we have lots of diversity in our surrounding. Anogesis latifolia, then આપણે ધવ અથવા ધાવડો કહીએ છીએ. So this Anogesis latifolia is also in our dry deciduous forest area majority. But we have Anogesis pendula, as I have mentioned earlier in the slide, that we have very good patches in the Sabarkantha Jessore Wildlife Sanctuary. Dr. Daraya is well known for this Anogesis pendula because he might have observed there. Then Anogesis sericea we have. So this is again third one, Anogesis species. Then we have Butea monosperma everywhere in the dry deciduous forest, but we have yellow colored Butea monosperma, that is variety lutea. So in some area you will find one or two this kind of trees of Butea having this yellow. ઘણી વખત લોકો એને કહે છે સફેદ ખાખરો. So that kind of khakra is also available, though in a very less number, but it is there, rare. Then see, this is Tinolepis garcini, and this Tinolepis garcini, see the structure of the fruit. It is slight oblong, but here I will show you Tinolepis, another species, which fruit you can distinguish from the fruit. See, it is round, it is siraciformis. So this kind of diversity we have lots of 5 to 6 Ceropegia species. Ceropegia pulposa, very common. Ceropegia candelabrum. Then these are the two, gokru. One is Betu gokru, other is Upu gokru. So one is Ziziphus. Sorry, not Ziziphus, sorry. Tribulus terrestris, that is Betu gokru. And other is Pedalium murex. So these two kind of gokra is available. Then we have very interesting this Rhytia species. One is Rhytia tinctoria and other is Rhytia tomentosa. See the different kinds of the pods. And we have another reported one species from the Jessore also, Dolichocarpa, Rhytia dolichocarpa. The length is more than 70 centimeter length of the fruit. So we have also observed this Rhytia, this dolichocarpa in forest of this Jessore Wildlife Sanctuary. This is slight different color, that is Tecomella undulata. Now I am concentrating on the indigenous community. See, generally લોકો એવું માનતા હોય છે કે ethnobotany. But it is very, very serious matter, that ethnobotanical study, how we can carry it out. This is not the discussion of the day, but this indigenous community have treasure of knowledge. And this treasure of knowledge having so many opportunity, and as a result we must conserve this indigenous properties or the knowledge. And this indigenous community, we will first see that indigenous this community which is spread in the India, as you just I don't think that you can observe this or not, because due to these very small letters, in India we have 8.6 ST population. Why I'm giving this population? Because most of the tribal peoples, they are residing in the forest area, and as a result they are utilizing nature and natural products which are based either on the plants or animal, whatever it is, mostly plant these products. They are directly utilizing, means they are in close contact with the nature. So that's why only Indian this ST population is given. In India it is 8.6%. See, these are the ethnic groups of India by state and territory. So if you just look at each and every state having this indigenous group or ethnic group. In Gujarat we have total 32 ethnic groups. Somewhere you will find some are omitted. So if we just remove 3, so at least 29 this ethnic groups are available in our area in Gujarat. So this having very good advantage. See, we have forest, we have different kind of ecosystem, we have different kinds of this plant species diversity, number one, and we have ethnic diversity in Gujarat is also available. So Gujarat is very lucky, and we are also lucky that we have this kind of both the diversity is available for the study of this ethnobotany or ethnomedicinal plants or indigenous knowledge. This is just census which show you that Banaskantha, Sabarkantha, then we have Panchmahals, then Dahod, some part of the Baroda, then Surat, then Valsad, Navsari. So this thing that you will see that we have lots of percentage of the ethnic group in each and every district. Just I want to interrupt. Can you observe this my presentation screen? Hello. Hello.
Yes, sir. Yes, sir. We can see. We can see your screen.
Okay. Thank you. Thank you, sir. Thank you, Dharesh. What is traditional knowledge? And sometimes, you know, people are confusing indigenous knowledge. So that's why I'm discussing the traditional knowledge, which is knowledge that which is held by members of a distinct culture and or sometimes acquired. So this is traditional knowledge which comes from a member of the distinct culture, and sometimes it is once you can also acquire, no problem. So this is traditional knowledge, that what we call traditional, conventional you can say, and indigenous knowledge which is restricted to a particular community or the society or the head of the society. Knowledge that is held and used by a people, group of people which identify themselves as a indigenous of this place. So indigenous knowledge which fits neatly in the traditional knowledge category, but all traditional knowledge is not necessarily indigenous knowledge. So this is the thing. So traditional knowledge and indigenous knowledge which is included in the traditional knowledge. And this traditional knowledge which is established on past experience and observations, it is usually a collective property of the society. Then it is, you see, it can be modified. It is not Ayurveda, remember. You can modify it as per your requirement and as per your knowledge. You can modify. There is no fixed thing. So it can be modified and it is enlarged as used over time. So means it is dynamic in nature and changes its character as the need of the people change, and it gain vitality from the being deeply entrenched in people's life, and they have very good faith. It is very common and quite straightforward that traditional knowledge on plants, especially the medicines we call medicinal plants, is generally available in their surrounding, affordable, and commonly used in large parts of the world, especially in Africa, Asia, and Latin America. Now I am discussing. I am really discussing indigenous knowledge. So what you mean by indigenous knowledge? See, indigenous knowledge is adaptive. Adaptive means it is based on historical experiences. Though it is historical experience, it is based, but it adapts to social, economic, environmental, not only that, but spiritual and political changes. So adaptation is the key survival of this indigenous knowledge. That's why it never vanishes. Then indigenous knowledge is cumulative. You know, so especially it is medicinal plants, suppose we call. So it is body of knowledge and skill that develop from the century of living in close proximity to the nature or the forest. So it is cumulative. Then it is also dynamic. It is not rooted, as I have mentioned, that it can be modified, no problem. It is not rooted in a particular point of history, but it has developed, it is adapted and grown over millennia. So it is not static, remember. Then it is holistic. All aspects of life are interconnected with this. So all aspect means cultural, social, then all this healing. Language is also included here in the indigenous. And nowadays, you know, indigenous language is also on the verge of extinction. And that's why this indigenous medicinal plant knowledge is also on the verge of extinction. So we have to collect the data before it vanishes. Then it is humble. Indigenous knowledge does not dictate how to control nature, remember. But it shows that how to live with a harmony with the nature and creators. Then it is intergenerational. The collective memory is passed with a community, means from father to son, like this. So from one generation to next generation, it is orally, it is distributed, or it is given through their language or stories or songs or ceremonies, whatever the part is there. With the help of that, they generally do. It is invaluable. See, sometimes you might have observed that we generally visit to the forest area and in remote area where some informants are there. Then we think that some people, they come to get their medicine, their ailment should be cured. So once they visited the very good and reputed hospital in the cities and metros, even though they are going there. So it is invaluable. It can be argued that indigenous knowledge, which is key to sustainable social and economic development, its growing recognition and respect for IK means intelligent, this indigenous knowledge, and a desire to collaborate with the indigenous community on environmental monitoring projects. It is irreplaceable. There is nothing Western science can do to replace it. So it cannot be replaced. Indigenous, as you know, as I mentioned, these languages are in decline. As a language die, so goes the indigenous knowledge also. Then it is moral. There is a morality in the indigenous knowledge, a right and wrong thing to interact with the nature. There is a responsibility given from the creator to respect the natural words, and that's why I've seen that once they dig out the root, so that they take what is required part, that much part they take, while remaining part should be again, it is dumped into its natural form. So that's why they have this moral, this duty, that we have to conserve the nature, we have to interact, we have to use it, but there should be a sustainable use of this. It is non-linear, means it is a circular, it is not straight. So as we know, different cycles are there, so it is cyclic. It is observant. Since its creation, indigenous leaders have observed their environment and made decisions for the community's well-being based on these observations. Then it is relative. Indigenous knowledge is not embodied at the same degree by all community members. Generally, elders will obviously carry this more knowledge than younger community, and now younger community they are losing this knowledge also. Then it is responsible. So indigenous peoples believe they are responsible for the well-being of the natural environment around them. So they always keep in a sustainable mode. Then it is spiritual. Indigenous knowledge is rooted in the social context that sees the world in terms of social and spiritual relation among the life forms, and it is unique. It is unique to a given culture or society. So there may be many similarity of this between communities, but generally it is unique for each community, and it is valid. It does not require any validation from the Western sciences, and that's why in remote area, when person having some illness, they generally consult the informants or Vaidya and get ready, then cured. So we have different herbal medicines, treatments, practitioners, informants, and they are main because source of primary health care, the care which is close to the home, accessible and affordable. This we have discussed also. Now, very important thing is there that prevention is better than cure. So when they are living with the nature and natural environment, then they daily use all these plants. So they remain always healthy. They will be energized, means having full energy. They have also immunity. Proper immunity should be there. So prevention is better than cure. And you know, the nowadays World Health Organization, we generally see the information given by WHO regarding COVID-19. So WHO which also indicate that more than half of the world's population does not have access to adequate healthcare services. So under this situation, those people which are residing in the village and remote areas, they could not afford this. They don't have access. So therefore, innovative alternative approaches or remedies are need to be addressed to this problem. And very important, again, very important part is the 80% of world's population, they are dependent on traditional remedies. So this is the thing. This is the photograph. If you just look here, that see how in the forest they are living very close to the nature. That's why it is given. And these are the list of the thing that all these drugs nowadays, what you observe, about 50 drugs we have discovered from the ethnobotanical leads, means from the indigenous knowledge or traditional knowledge by this pharmaceutical company. So we have again lot of potential to develop a new drugs. So there is a great scope for new drug discoveries based on the traditional or indigenous plant uses. And during this one recent survey, 8,000 wild plant species were recorded, and out of which 2,000 are found to be new claims and worthy of scientific scrutiny. And see, this intellectual property, so far as the intellectual property for the indigenous knowledge is concerned, CBD is always mentioned. There are 3 goals of the CBD as per the Article 8J. We have to respect, preserve and maintain indigenous biological knowledge. We have to promote the wider application of traditional knowledge and to ensure equitable sharing of benefits. So this is the requirement, that we must preserve this indigenous knowledge. We must disseminate this in a wider area, and so the people must maintain their health and immunity. This healthy wins that I don't want to discuss. This is one Arogya Pacha, that is green, that gives strength. Trichopus zeylenicus belongs to Dioscoriaceae family in the Kerala, where Kanni tribe is living. So this is again a history, that this plant, during this field trip, some of the scientists, they feel that why this all these tribals they are getting energy. So once during this roaming in the field, they take this fruit of this Trichopus zeylenicus, and from that they have developed one Jivani. See, this is the Jivani, and through this Jivani they have added some other ingredients, and finally they have developed a model, that is benefit sharing. And this benefit sharing model, for that Dr. P. Pushpagadan received a UN Equator Initiative Prize in 2002. Dr. Pushp became the first scientist in the world that experimented benefit sharing model. We call it a TBGI model or Kanni model, on the basis of the name of the Kanni tribes of the Kerala. So this is implemented in our India also. Now, see, when you are going for the this recording of this indigenous knowledge, remember that information on traditional knowledge of plants can be gathered at any time, whenever research associated with the users, means informants. No need to ask for any or many questions to the informants. Give respect to the tribal informants. See, I'm giving here. I'm giving here a list of some of the things. Some of the things in the sense that for one particular disease, for the treatment of this disease, see, I'm giving some clips or video here. Let's see, in different area, different informants using different plants. This is the beauty of the indigenous knowledge, and that's why we have to collect first this indigenous knowledge. Here, say, sometimes some people, what they believe, that what is use of this? Any people can give this information, but it is not like this. Then once we get this indigenous knowledge and all this recipe, then this can be shared with the pharmaceutical person. And once we share with that pharmaceutical person, then we come to know whether this treatment is actually it's working, this plant is working or not, means we can say about the efficacy of this plant. So here I have first I have selected asthma. Sarji, just you just tell me the time, okay? When I have to finish, please.
Okay, sir.
Otherwise,
Okay, sir.
otherwise I will continue. Don't worry.
Okay, sir. Okay.
How many
Sir, you can share up to
minutes are
10,
left now?
you can share up to 10 minutes, sir, and later Dr. Panchal also will join for 10 minutes, and after that, Dr. Maharaja Chowda, Honorable Vice Chancellor, also joined us with us, and at
Okay.
last he will do the presentation lecture, sir. Please, thank you.
Okay. Thank you. So I will continue. Say this Mansukoi Dabi, which is there in the Barnet Gir forest. See, this fellow is using Scabiola takada. The plant is also on the screen. So the leaves are used to treat the asthma. And says, listen, in Gujarati it is there. So other than Gujarati people will not understand. But this leaf juice or decoction which is given to this patient of the asthma, and this patient can be cured within this 15 or days or 1 week. So this treatment for kindly please listen this. If you try to understand this.
One time પાણી પીવે 15 દિવસમાં સાહેબ હું એમ કહું છું કે
within 15 days, not only walk, so asthma
કેટલા દિવસ પીવાનું ઈ તો પછી એના અનુકૂળ 15 દિવસ પીવું પ્રેમ થોડી કપ
this is, you know, the Scabiola takada generally the people give as a Rudraks, but it is not Rudraks, not pure Rudraks, it is
ઈ તો હું કહું ક્યાં તમે સારી માથે નર્મદ આપણે હા બસ ઉપર એમને પૂછ્યું સારીમાં કઈ જગ્યાએ લોકેશન મને કહેજો હું પેટ્રોલ પંપની પાછળ પેટ્રોલ પંપની પાછળ શિવ મંદિર પેટ્રોલ પંપની પાછળ શિવ મંદિર હા અને આખું પાન કેવું હોય આખું લીલું કમ્પ્લીટ આવું ફળ ઝાડ હોય મીઠી લાલ હોય
Unmute. Super. Punjani sir, please unmute yourself. Excuse me, Punjani sir, can you hear me? Please unmute yourself, sir.
Honorable Vice Chancellor,
Please
Dr.
unmute yourself.
Anjana, please. Our Honorable Vice Chancellor, Dr. Maharaja Chowda, also joined us webinar at last, and he will address. So, Punjani ji, so please be continue your presentation. Our Honorable Vice Chancellor is saying your presentation also, and after that, as per indicating time, he will address our community of botany, zoology, and microbiology. So, Punjani ji, please be continue.
Punjani sir, can you hear me? Please unmute yourself. Can you please unmute yourself, sir? Punjani sir. I think there has been some issue again.
Dear friend, Punjani sir has explained a very good indigenous knowledge about the plants. Yes, sir.
Yes, sir.
So,
Yes.
muted? Yes, we can. Our Punjani ji, our Honorable Vice Chancellor
Hari.
also observing this. So please continue the indigenous knowledge about the plants of tea. Please, thank you.
Welcome, Honorable Vice Chancellor ji. Namaskar, namaskar. We are presenting. Madam, can you see the screen? Presentation screen?
Yes. Yes, but please full screen. Please do full screen.
Done?
Yes, fine. Very nice. Yeah, done.
Okay. 1 minute, please. 1 minute, please. Now see, we will again see another clip where the Naranbhai Chavda from this Parada Hill area is also treating asthma, same disease, but with different kind of plants. So this is variety. What we get this variety, what we get the diversity, and the same people, same Naranbhai will also treat the asthma with another plant in this area. See, this for asthma, Puraria tuberosa, which is used by this, the root is used.
Vidarikan.
This is Vidarikan.
શ્વાસ જેના કણનો દૂધમાં નાખી ઉકાળો કરી અને સવારમાં ભૂખે પેટે એને પીવાનું અને 20 વર્ષનો શ્વાસ 20 દિવસમાં ચૂપ એટલે ચૂપ.
Means this can be cured within 20 days. See.
નૈણા કોઠે દરરોજ એક દિવસ દરરોજ
Daily you have to take this.
શ્વાસના દર્દ મારે કઈ અંચોક આવી ગયા હા અને બધા રેગ્યુલર જૂનામાંથી ઉભા ન થઈ જતા હોય હળિયું કાઢવા પડતી. વાહ દાદા વાહ.
Means can walk, no problem.
બસ આ કામ કરવાનું છે બધાએ.
See, the same person with treating this asthma with the help of another plant, that is Dichrostechus cinerea. So see how he is giving the fruit powder.
દમ માટે કામ કરે દરરોજ સવારે. હા દર ભૂખે પેટી સવાર આનો પરિણામ આપી દઈએ. દમ મટી જાય. હા કઈ વાત હતું. મોર ડોન્ટ બરાબર.
Moodwadiya Sahib is there. Okay. Now see, in the Sabarkanta Biloda, the same disease ailment, that is asthma, which can be cured with the help of only inflorescence of the Echinops echinatus. Utkantara.
ઉપર સુધી મૂંગો બંધાણો છે ને
કઈ વનસ્પતિ?
આ ઉટકોટ કહેવામાં આવે છે. ઉટકોટ કહેવામાં આવે છે. એ શ્વાસ પદાર્થ છે ને એકદમ બરોબર. એમાં આ ડોડી છે ને એ આપણે દેવતાથી બાળી મૂકવાની હેડી બને ને એ બે વખતે પકડી હવારમાં લેવાની. હા
કેટલા દિવસ પીવડાવવાનું?
7 દિવસ. ના કરશું. એ શ્વાસ ક્રિયા.
વાઇસ ચાન્સેલર શ્રી આવી ગયા છે, મેં જોયું. અમને નમસ્કાર પણ મેં કરી દીધા છે. See, okay. See, the person that is Ninama Sauji bhai is treating asthma with the help of one plant that is Viscum articulatum. જેને આપણે ગુજરાતીની અંદર એને કહીએ છીએ વાંદો, પાંદડા વગરનો. અને એ લોકોની લેંગ્વેજમાં, લોકલ લેંગ્વેજ, ઇન વર્નાક્યુલર લેંગ્વેજ, they call this plant as a Timberwano borico. So this asthma can be cured. But not only asthma, but arthritis can be cured also. Remember, with this plant Viscum articulatum. So let us listen to this Ninama Sauji.
Punjani sir, sorry for interruption.
Okay, sir.
Hello, Punjani.
Haji, bolo.
Please be focus the screen for only your face. I think, yeah. Please be focus your screen for only your face. Please. Yeah, fine. Thank you.
So my presentation is not there.
presentation is coming, but only your face is actually comes with another people also. Please be focused screen for only your face. Thank you.
So another people is sharing this presentation. He has to stop it.
એક કુકી નાખીએ આ કેટલું લેવાનું કેટલું છે
So I think
Could you please make your background clear? Only you should be.
hello.
ઘણી જડીબુટી છે
Sir, can you hear me?
આમ ખાસ
Stop. Can I stop now?
guaranteed કઈ આમ એમાંથી
Sir, you have 4 minutes left now, so you can just
Can
finish
you
your talk.
can you see my presentation on the screen?
Yes, sir, it is there. Please continue, sir. 4 minutes are left.
Okay. Just now I'm doing. Okay.
એમ
I'm finishing, please.
એ મધુરી કપૂરી છે એ કમરામાં કામ આવે ને
This is Madhuri Kapuri, which is used
Kamrani
to
fish.
treat the kidney stone.
પછી વિક્રો છે એ કમળામાં
વિક્રમ
પછી ભો આપણે ભોઆંબલી છે એ કમળામાં કામ
બરાબર
ભોઆંબલી પતરી ભરી કામ કરે
અચ્છા અચ્છા નહીં આ
બાકી કારી જોરગોળી છે
હમ.
એ પતરીને ખાસ પતરીની ગુળ લાંબા ચીરની ગાળે દુખાવો ન થવા દે
કેમ નિર્ગુંડી
હા
એટલે
કાળી જોરગોળી
એ વળી કઈ આવે
મોટા તમે એને નગોડ કહેતા હે
આ નગોડ ખરી બરોબર સંસ્કૃતમાં નિર્ગુંડી કહેતા હોય
હા
એ નગોડમાં તો બે જાત થાય છે ને 3 પાંદડા વાળી ને 5
Okay, so now stop. I stop it.
Okay, thank you very much, Punjani ji. You actually delivered very good knowledge about the plants, which actually found indigenous knowledge about the plants, which are found in the rural area as well as the forest area. One thing also, sir, you has explained that adaptations, when we are facing against the COVID-19 this scenario, the human beings have to find out this very perfect solution in front of the COVID-19 right now. So that also you explained very nicely that moral levels and non-linear levels and relative knowledge regarding these things also, responsibility as well as the spiritual reality of the plant. In the real sense, we delivered all 3 lectures regarding the ethnobotanical knowledge that you shared a lot, and participants actually enjoyed these things. Our Honorable Vice Chancellor also available today.
Yes, sir.
But before that, I am requesting to Dr. Rakesh Panchal, Honorable Vice Chancellor Dr. Maharaja Chavda is available here in our webinar. Dr. Panchalji, I am requesting to you that you are indicating in your presentation that the industrial level drug manufacturing. So our Honorable Vice Chancellor also eager to know about this thing. That's why we are giving to 6 minutes to you, sir, and rather than
So nice.
Vice
Sir.
Chancellor
Thank you.
will
Thank you very much. Rapidly, I will go through.
this thing. Yeah.
Rapidly, I will go through this. Very fine. Finish. Try to finish. This is what where we stopped here. We are manufacturing the protein products. They are inside. Microorganisms are to be grown inside, and then after the protein is to be purified. Okay. Next one is when we are growing the fungi, that is Aspergillus niger, used to grow fungi in 100 ml broth medium on shaker for 48 hours, means 2 days, and it forms spherical pellets. These spherical pellets are vegetative form of fungi, and these pellets used to be transferred to fermenter. So when you grow the pellet, whether it has been grown well or not, that is to be seen under microscope. So all these pellets under low power microscope are looking like a sphere, and this sphere are having central compact mycelia and peripheral mycelia are coming out. This peripheral mycelia are absorbing the nutrient, and then central mycelia process it and produce the product for us. So the same thing under high power objective, that is looking like a tubular structure. This tubular structure are absorbing nutrient and then producing product for us. So we need to maintain the manufacturing process in a way where we require the cloning and selection of the organisms. Gene of interest is to be transferred to the vector, and then after vector is to be transferred to host, and this host is to be grown in fermenter. So So where you are growing host in fermenter, at the time all the fermentation parameters are to be controlled. And in this case, various hosts are to be collected by us, and we are keeping it in form of a cell banking. And this cell banks is having various kind of organisms which are capable to produce various kinds of products. So company do have its own cell bank, and there are national collection centers are also available, international collection centers are also available. So we can choose an organism from that, then engineer it, and then after produce the product. Of course, during manufacturing, the warehouse input and output is also to be controlled. Where manufacturing is to be done in industry, we do have two set of process. One process is upstream processing. The second one is downstream. In upstream, what we do is we are producing the product of interest, and in downstream, we are recovering the product of interest. In producing the product, we do have the requirement of media, then inoculum, the culture which I have shown on the flask. Then after the seed generation, after that, it is transferred to the fermenter, which I have shown there. In fermenter, all the parameters are to be controlled. And of course, the nutrients are to be added. Of course, nutrient must be the cheaper material, so as your manufacturing cost may be reduced. And if your product is intracellular, then you need to kill the cell, break open the cell, and recover the product. If the product is extracellular, then problem is not that, because it is secreted outside the organism, and then after we just filter the organism out, and from the broth we can recover the product. And where the product is to be separated, the centrifugation is the best. Some cases we may use filtration also. In case of downstream processing, we require chromatography, maybe a kind of flow, and all the things are controlled so as it can be separated on the basis of organic solvents which are to be added. We may have diafiltration, we may have ultrafiltration, and once the product is separated, we may purify the product, and formulation is done. Of course, it is to be filtered through 0.22 micron filter, so as it can be sterilized at the same time, and it is to be stored at low temperature till it is being packed and transported. Filling and packing also requires expertization, and ultimately it is packed and dispatched to the market. But the production is conducted with the waste material, because when it is a biological product, the product is formed at the same time, numbers of byproducts are also produced, and all these byproducts may not be useful, and these byproducts are creating a lot of contamination, pollution of environment. So waste handling and disposition of waste, this is a big headache for the industry, and that is how industry people are always looking for pollution control board people, and all the time they are pleasing there, so as the closure notice may not be there from the governing authority. So waste handling and waste degradation, waste disposal is a big task for the industry. Where you go for analytical microbiological testing, you need to have a lot of testing like purity, the impurity is to be detected, then charge variants are to be done, concentration is to be worked out, bioassay to find the efficacy of the product, whether it is antibiotic, it is going to kill the bacteria or any other microorganism, we'll have to test it there in form of bioassay. After all this, we need to have review the data, and stability of the product is to be studied, the real-time stability as well as accelerated stability is also studied, and this stability study will give you idea about the shelf life of your product, and this shelf life is to be denoted on the product. So all this kind of job is also an additional job which has been done in the industry. Everything is to be validated in industry, like process validation, cleaning validation, software validation, equipment, method, filter, everything, whatever you are using, all the steps are to be validated. And if at all you are going for this, you'll find that the product which has been approved by the competent authority in India or maybe worldwide, if you go through the approval of 1982 to 2008, you'll find that the biological product, only one product has been approved in 1982. Later on, the number of product has increased. It came to 2008, as many as 30 biological product has been approved by the competent authority. And if you go to 2019, you will find that more than 150 biological product had been approved by competent authority worldwide. So you can produce the product and sell it in the market in form of medicine. The next coming is a biosimilar. What you call is a generic drug. The generic drug basically means that it is a chemical drug manufactured by chemical process, and biosimilars are the biological molecules which are similar to the original innovator molecule. But this similarity is not 100% identical. So when biosimilar is played, we say that biological medical product, which is almost an identical copy, we call it as almost an identical, it is not identical, right, to the original product. So original product patent right has expired, and after expiry of patent, the same molecule can be manufactured by any of the company across the globe. So after expiration of patent, you are trying to produce the molecule and sell it in market. Here, the main advantage is you need not to go through all kind of testing on animal, testing on human, because all this job has been done by original innovator, and lot of time and money has been spent after that. But that all benefit you are getting, so you need not to go for all this test, and only to produce the product to show that your product is similar to the original innovator product, and you can market it. There are certain agencies which can go through the bindings that Europe, Middle East, and Africa, EMEA countries approval is to be done. The first molecule approved by EMEA, that was in April 2006. These were the first in world. European people are the first in world to give approval to biosimilars. So first biosimilar approved in 2006, it was Omnitrop, which was been similar to genotropin. So genotropin, a kind of somatostatin growth hormone. So the patent has expired, and then after the patent has expired in 2001, and then after the first biosimilar came in 2006. So 5 years time the company has taken to understand the molecule and to produce the molecule. One more is Valtropin. The Valtropin is a generic molecule that is called biosimilar molecule, and it is biosimilar to Humatrop. So original company manufactured Humatrop using Escherichia coli bacteria. So using this bacteria by fermentation, they were manufacturing Humatrop. And the biosimilar manufacturing form of Valtrop, that was using yeast. Yeast is a eukaryotic unicellular organism, and the bacteria is prokaryotic unicellular organism. So organism changes, and when organism changes, you say that lot of change has been there in the molecule. So molecule by its effect may be same, but it is not exactly identical to the original one. So this molecule may have side effect. So this is what the problem is. Indian scenario of biosimilar, if we talk about, we'll say that similar biologic, this is been there in 2000. So we have approved the hepatitis B vaccine, HPV vaccine, that was according to GABI, it was been approved, that is generics and biosimilar initiatives. And this molecule has been a boon at the time where it was being marketed. So we'll see slight difference between generic drug and biosimilar. We'll say that generic drug are chemical. It is a simple molecule. To understand the molecule is very easy, whereas the biosimilar are slight complicated molecules. So understanding is a little difficult. So you'll find that the structure of this generic drug is smaller and it is less complex, whereas structure of biosimilar is a large one and it is a complicated. You can say 100 to 1,000 times complications to the generic molecules here, since it is a biologic. Molecular weight is very low, as you have seen in case of aspirin, it is 180 Dalton only, and here in biosimilars it is very high. In case of growth hormone, it is about 5,008. In case of antibodies, that is monoclonal antibody, it may be 150,000 Dalton. So it is quite complicated. The generic molecule are more stable because structure is simple and all the manufacturing is exactly identical to the chemical. Whereas your biosimilars are very sensitive to changes. As I say that you are producing product using another organism and the product is sensitive for the changes. So physical changes may be there, the chemical changes may be there. If you go through generic drug, you'll find that route is oral. By oral administration, you will find all different kind of capsule and tablets which are generic. You can use oral route for administration. Whereas in case of biosimilar, by and large, the route is injection as well as inhalation, all kind of biosimilars. The manufacturing process is less complex in case of generic, and it is more complex in case of biosimilar. The registration is also simple. If you go through the generic drug, you need to register to an ANDA, and if you go for the biosimilar, you need to go for European and Middle East, Africa agency, as well as Food and Drug Administration. This is for USA, Food and Drug Administration. Whereas in India, the things are slightly loose so far as the complex structure and approval is concerned. So far as biosimilar is there, you need to get the licensing, and where the licensing is done by European people, the European people are going for the similarity of the original reference product. So original reference product and your product should be similar. And another one is meaningful differences should not be there. There should not be any difference in quality, there should not be any difference in safety, and there should not be any difference in efficacy. So quality, safety, and effectiveness should be the same. Then that molecule is passed by European people. For example, I say that this is your original molecule, which has been invented by the inventor company, where the biological active part may be same in case of your biosimilar, just like as the key. But what about the holding portion? Holding portion of key may be different. You say that non-operative part may be different, and this non-operative part may be the responsible part which can generate the kind of allergy, which can generate the kind of side effects. So one molecule original inventor has gone through all kind of testing, has invested a lot of money, and the same molecular manufacturing biological active part considering only. So effectiveness is 100% same, but it can have side effects. And that is how the cost is low. The people has to decide whether to work with the efficacy or some side effect can be tolerated, but effectiveness is the same. So this kind of molecules are utilized by people. So far as biosimilars are concerned, you'll find that the biosimilars are a biological product. It is highly similar to reference product, and there should not be any clinical or meaningful difference from the reference product. No difference should be there so far as clinical efficacy is concerned. And the approved biosimilar, that is by Food and Drug Administration USA, and it goes to a rigorous testing. All kind of testing is done. For manufacturing of biosimilar, what you do is you have to isolate one gene from the genome. That gene is transferred to the vector, a vehicle. Then after that gene through vector, it is transferred to the cell which is going to produce product for you. After that, it goes to a fermenter.
Panchal sir, sorry for interrupting. Could you please conclude your talk within 2 minutes?
Okay, 2 minutes. Finish. Okay. I'll skip numbers of slides. Okay. We'll come to the last stage of this. A lot of biosimilars are being approved by Indian government also, and these biosimilars, there are numbers of agencies for approval in India, but the problem in India is the Indian control that is not there, and that is how numbers of multinational companies are coming in this country for manufacturing as well as selling. I'll go to last slide because a lot of huge money involved in this. But the major players in India for biosimilar manufacturing is Dr. Reddy, Biocon, Intas Pharma, Ranbaxy, Glenmark, Reliance, and Biology, as well as Pharma in Ahmedabad. That is also working similars there. There are a number of products there in biosimilar. In this regard, in my laboratory at Gujarat University, Department of Microbial Biotechnology, we are also working on 2 molecules. One is streptokinase, which has been the molecule which is used at the time of massive heart attack. The people used to inject the drug, and this drug remain in body for 1 day, and finally the heart congestion, heart clot, that is dissolved, and the person's life is saved. So streptokinase is been done by one of the major PhD research by Bridges, and staphylokinase, another same kind of molecule. Efficacy wise, it is the same, but the binding capacity is slightly different. It is staphylokinase, which is also done by another research fellow from Iraq, Mr. Barkat. One more molecule we are manufacturing in our laboratory so far as the process is concerned, beta carotene and lycopene. These are very important molecules to be used in cancer therapy, where one more leading PhD is working in our laboratory on this beta carotene production. We are using the bacteria which are from the halophilic environment. Another one which we are producing is the cellulases and producing second generation bioethanol. This is in environment protection. One more molecule we are manufacturing, that is the plant growth promoting hormone, that is gibberellic acid using fungi. And another one is the environment pollution control in form of cellulosic industrial waste degradation. This molecule production, that is also there in our laboratory, which is at Gujarat University. The number of controlling agencies are there in India. And at last, I thank all the organizers from Bhavnagar University, particularly Professor Bharat Singh Gohil, and all my other friends from the Department of Life Sciences, Bhavnagar University. Thank you very much.
Thank you so much, sir, for your lucid presentation. Honorable Vice Chancellor Maharaja Krishnakumarsinhji, Bhavnagar University, Dr. Maipat Singhji Chawda, whose dynamic leadership is bringing several reforms to the university, has always been a source of inspiration for us to organize seminars and workshops. May I now request Honorable Vice Chancellor, sir, to please deliver his presidential talk. Please, sir.
Good afternoon.
Good afternoon, sir.
Good afternoon.
Welcome to speakers, participants, and dear students. I am happy that Department of Life Science has organized a webinar on spectrum of life sciences, which is covering various aspects of biological sciences. I am sure that more than 850 participants who have registered will enrich their knowledge. I appreciate the speakers, Dr. Rakesh Panchal from Gujarat University, Dr. Nishitariya and Dr. Bhaskar Punjani from Hemchandracharya North Gujarat University, Patan, for sharing their research and views on different fields, including affordable drug development, research opportunities in less explored fields, and indigenous knowledge about plants. अभी मैंने ज्वाइन किया, पुंजानी साहब का प्रेजेंटेशन चल रहा था, बर्डा हिल का, फिलोडा का भी प्रेजेंटेशन देखा, और अलग अलग प्लांट, हर्बल प्लांट पर, चाहे गुजराती में हम कहते हैं विकड़ो, नगोड़, चाहे किडनी स्टोन हो या अस्थमा हो या आर्थराइटिस में ये जो प्लांट का क्या इंपॉर्टेंस है या तो क्या रोल है, इसके बारे में प्रेजेंटेशन देखा, बहुत बढ़िया रहा. Since the inception in 1985, the Department of Life Science is actively engaged in academic research and extension activities. The department is offering PG programs in botany, microbiology, and zoology, and PhD in zoology. So far, the department has produced over 130 PhDs. The department has received research grants from various national funding agencies like UGC, CSIR, and DST. The department has also run Earth Science and Technology Cell, sanctioned by Ministry of Earth Sciences, Government of India. The department has a very sophisticated instrumentation facility. The department has contributed over 500 research papers, several books and monographs in the subjects botany, microbiology, and zoology. Research scholars of the department have received various national and state level fellowship like Inspire Fellowship, Maulana Azad Fellowship, and Shodh Fellowships. Further, research scholars, PDFs, and faculties of the department have received international travel support awards as well. Recently, the department had organized an international conference on panorama of life sciences, which was participated by 600 participants, and 450 participants presented their research papers at the conference. This webinar also will be very interactive and informative to students, research fellows, and faculties, and there will be some constructive outputs in this difficult time. I wish the webinar all success. I congratulate the convener of the webinar and his team for organizing successful webinar. Thank you. Thank you very much.
Nancy Gohil for concluding remarks. May I now invite Dr. Bharat Singh for the concluding remarks.
Yes, thank you. So it is warmly information today shared by the very expert speaker by the different subjects of life sciences, botany, zoology, and microbiology. Sir, today we are very happy to say that Panchal sir has actually explained very recently topic which we are facing against the COVID-19, and he has delivered very nice information regarding the drug manufacturing. And another speaker was Dr. Nishitariya. Sir, he has also explained very well the lesser mammal study. What we have the heritage of in India, the fauna, flora, and all, the life sciences, the science about the life actually the dealing the very informative lectures today. Dr. Daria has also told that it is also very important study should be there in each and every universities which are not actually studied. It is not under the data that we have to collect. And third speaker also has explained very well that the without plant, the indigenous knowledge and rural knowledge of the medicine, he has explained well. And we have to actually encourage the knowledge which actually found in the rural areas regarding the plant knowledge. And according to ethnobotanical knowledge he has shared. Sir, in the real sense, the umbrella of this subject in the Department of Life Sciences shared very nicely all three speakers. And a part of my sides, we are very much thankful to all expert speakers in this webinar Spectrum of Life Sciences. Sir, really he has actually make the lighten and depth of the spectrum of life sciences, all three subjects. And sir, your valuable presence in our webinar, Dr. Maharaja Chawda, Honorable Vice Chancellor, he has also joined it today and warm us. Sir, we are very much thankful to you also. And spectrum of life sciences really today lighten. Thank you. I am requesting to Dr. Anjanawala, please take the word of thanks last minute, please. Thank you.
Thank you, sir. As we are concluding the webinar, before moving on to vote of thanks, I would like to make one announcement. The participants may please note down the screenshot which will be displayed shortly, which gives the email addresses of the speakers, distinguished speakers. Whosoever has the queries regarding their respective topics, they may please direct through email. So shortly it will be displayed, the screenshot you may have. So as we are concluding the webinar, on behalf of the Life Sciences Department, I would like to deliver vote of thanks. At this outset, our sincere thanks to our Honorable Vice Chancellor, Dr. Maharaja Chawda, for his continuous support and encouragement, without which this event could not have been possible. I must mention our deep appreciation for our distinguished speakers, Dr. Rakesh Kumar Panchal, Dr. Nishitariya, and Dr. Punjani, for sharing with us their valuable findings and views. Our special thanks to Professor Dilip Barad, English Department, for his continuous support and providing us with the infrastructure. Our thanks are also due to our Registrar Maharaja Krishnakumarsinhji, Bhavnagar University, Dr. Kaushik Bhat, and University Administration for their kind support. At this outset, I would also like to acknowledge the media for covering the entire event, and staff of Life Sciences Department also is gratefully acknowledged. I must mention the acknowledgement of Ronak Bhai, who has been technical support throughout the webinar. Last but not the least, the research scholars and the students of life sciences who have been the backbone for this webinar, and especially I must mention name of Naina Dabi, who has worked I think very much for last 1 month, and directly or indirectly whosoever has helped for this webinar, I thank them one and all. Thank you so much. Have a good day, stay safe, stay healthy. Thank you so much. We are just displaying the screenshot, so just note it down, please. Chennai, where is Chennai? Okay, thank you all. Thank you so much.
Thank you. Thank you.